Good morning. Welcome to this BroadE about doing genetic screening with the GPP. I'm John Dench. I've got quite a lineup of speakers today that sort of are intended to cover every aspect of the screen that you might do, starting with conceiving, should I do a screen, how do I pick a good model, how do I pick a good assay, through execution of the screen, and then how to analyze and follow up from your screen after that. So we're scheduled to be here from 9 a.m. until 1 p.m. We're going to take a break about halfway through, but obviously, if you can you're free to get up and leave at any time. There's a Keurig coffee maker right out in the lobby there. If you're not familiar with the BroadE, there are restrooms both if you go down the hallway that way towards the labs, or if you go that way past the other conference room, there are some restrooms. All the slides for today are available for download, so if you want to follow along and take notes on the slides themselves, here are the links to them. They're also written up here and over there, so if you want to follow along and take notes, you're welcome to do so. Okay, here's the outline of the course again. You can hear from many speakers, and again, a 10-minute break somewhere in the middle. Okay, so the sort of reason to exist for the GPP is that our genome is kind of a dumpster fire of information. So imagine you went to IKEA, and you bought 20,000 things representing all 20,000 protein-coding genes in the human genome, but then mixed all the parts together and burnt the instructions. So then sitting on your living room floor would be all the parts of a Klondorp and a Flopidip, but you had no idea how to put it together, and you just had a lot of Allen wrenches. And the reason, for why it's so complicated is that the genome is the product of 3.5 billion years of short-term thinking. It's obviously was not top-down, rationally designed, where all the genes that do this one thing are all right next to each other, all the genes that do this other thing are right next to each other. It's just kind of a complete mismatch. And of course, the reason for the BroadE was kind of, well, what are all the genes, the Human Genome Project? So we've had the sequence of the Human Genome Project for quite some time, but of course, this in and of itself is not terribly informative. Or as Eric Lander once summarized it in 7 words, Genome bought the book, hard to read, and if you want to read it and you're a little confused, you can't just call up some helpful people in Sweden to tell you what to do. Okay, so how do you determine function? How do you go from something like this to then say, okay, well, if I have a variant at this nucleotide and I have a 2% higher chance of developing diabetes at some point in my life, how do you go from sequence to function? Well, obviously, the classical way of doing it, so what's called classical reverse genetics, is you start with a phenotype. So, for example, you eventually find a Drosophila that has a white eye, and then you map backwards to find the gene that's involved. You have a family that has a predisposition to cancer, and you map backwards to find the BRCA1 gene. But of course, now that we have the sequence of so many genomes, what we want to do is forward genetics. We want to start with the sequence of the gene, and from that, determine the phenotypes that gene might give rise to. Now, not long ago was a pretty dark time in functional genomics. So this is a quote from Bill Kalin, our colleague at Dana-Farber. So he wrote an article in Science about the use and abuse of RNAi to study mammalian gene function. And he started off the review by noting that the honeymoon is over. Now, if you're married to a technology or a person, you don't want to hear that. And he noted that the big reason for why RNAi had fallen so short of expectations was off-target effects. So RNAi, and GPP was originally founded as the RNAi platform. The issue with RNAi is that when you put a small RNA into cells, it can do 2 things. It can do the thing you want, so you can put in a small RNA, and it can load into the Argonaute protein complex, also known as RISC, and it can bind to a messenger RNA with 18 to 20 nucleotides of specificity, cut that messenger RNA, and great, you've achieved successful knockdown of your gene of interest, and you can study the cells. However, a small RNA can also enter the endogenous microRNA pathway. And when a small RNA enters the microRNA pathway, it binds to one of several Argonaute proteins, not all of which can actually cut messenger RNAs, but it binds with only 6 to 8 nucleotides of specificity. So if you only have 6 to 8 nucleotides of specificity, obviously you're going to bind to many, many different transcripts. So this is what we want to happen, this top pathway, but this bottom pathway also happens anytime you try to do an RNAi experiment. And you can either ignore that, which is somewhat what the field did for many years, or you can come to grips with it. So as an example, in using gene expression data, we asked, and this was in collaboration with the connectivity map, We asked, okay, well, if we put in two different shRNAs that both target the same gene, in this example, BRAF, or we put in an shRNA that shares this same seed sequence, the same 7 nucleotide region that triggers microRNA-like activity, and we asked, well, which of these two are more similar when we put them into cells? And we didn't do this with just one set of shRNAs, but with thousands. And what you see is that, okay, what's plotted on the x-axis of this histogram is Spearman correlation. How similar are any two shRNA signatures? And if I get close enough, I can actually fit my laser pointer in between the null distribution and the pairs of shRNAs targeting the same gene, but obviously there's not a lot of signal there. However, any two shRNAs that share the same seed sequence, the green line here, are much more likely to show a consistent Spearman correlation. This suggests that the off-target effects of RNAi is most of what's happening when you put them into cells. And this is why, when CRISPR technology came around, we were so excited about it. It offered a different way of determining gene function. So the CRISPR system, we're not going to get into the history or the details of how it was discovered or anything like that, but suffice it to say, it's a two-component system in mammalian cells. You need two things. First, you need a protein called Cas9, and second, you need an RNA. And if you put that RNA and that protein together, and you put them into cells, then they trigger a site-specific double-strand DNA break. And in mammalian cells, if you have a double-strand DNA break, what usually happens is the non-homologous end-joining pathway recognizes this double-strand DNA break and repairs it in a way that is somewhat sloppy. So you tend to have either an insertion or deletion of a small number of nucleotides, which, of course, in the vast majority of the genome, in indel probably won't have too much function, which is why when our DNA is being cut constantly, as it is, and we repair by NHEJ, we're still fine. But if you target it to a protein coding region, then you can often engender a frameshift mutation and thus a null allele. So the simplicity of programming CRISPR is why it's become so widely used. Again, this is just a zoom in of the RNA. Basically, the first 20 nucleotides of the RNA can be programmed with essentially any sequence you want to dictate where Cas9 is going to bind in the genome and deliver a cut site. So, with just 20 nucleotides of specificity, you can cut kind of anything you want in the genome. And then when we look in gene expression data, these are the same data sets from the connectivity map that I showed previously, where we plot either off-target magnitude on the y-axis or on-target magnitude on the x, and each dot here represents a different shRNA on the left or a different sgRNA on the right. You can see that with RNAi, there is on-target magnitude. Most of these dots are shifted to the right of 0. They do have an on-target magnitude, but the off-target magnitude is much higher. So this is completely consistent with the correlations that I showed previously. However, with CRISPR technology, the vast majority of the dots are below this x equals y line here, meaning that the majority of CRISPR perturbations have more on-target magnitude than off-target magnitude. So when you have a reagent and the tube says, okay, this RNA targets this gene, with CRISPR you can generally believe that annotation, whereas with RNAi, if you're relying on a single shRNA to determine your phenotype, you're often going to be misled, which is why with RNAi and with CRISPR technology, but much more so with RNAi, it's really critical to rely on multiple different perturbations, all of the same sequence, to give you the same phenotype before concluding that gene was really involved. Okay, so how does the GPP actually work? And this is what today is going to try to get at. You have an idea for a genetic screen. You think you want to do a screen. How do you actually interact with the GPP? So the way we view it is that the goal of the genetic perturbation platform is to build good perturbations and to really understand how they work, what their strengths are, what their weaknesses are, and to accumulate a collective wisdom of why do screens fail and what are the characteristics of screens that succeed, such that if you're doing your first screen, you can learn from that collective wisdom and not reinvent the wheel. Whereas the job of the collaborator is to build good models. The job of the collaborator is to understand their biology terribly well. I know this much biology about this many topics. Don't rely on us for expertise on diabetes, expertise on cancer, expertise on any one area of biology. That's what you guys are focused on. But then when we come together, so the first step is we meet to discuss the screen. and we provide advice. We ask a lot of questions. And then what we each try to, the way the model works is we're focused on what our strengths are. So in this example, we would provide the perturbations. If you want to do a genome-wide pooled screen, we would provide, say, lentivirus, so that you don't need to learn how to grow up libraries and maintain representation. You don't need to learn how to do lentiviral preps. We just have gallons of lentivirus sitting in the freezer ready to hand out. Likewise, you then execute the screen. You are an expert in handling your cells, let's say, or handling your mouse model. It does not make sense to try to transfer that expertise to us. It makes far more sense for you to execute the screen in your own lab. And I should point out that, the way this works is not unidirectional, but to have a lot of back and forth during the screen itself, whether that's by email, whether that's in person. I have several collaborators who aren't at the BroadE, who are, in California or St. Louis, and I've never physically met them, but we've done many screens together because we have a constant back and forth on the phone and over email to just figure out, how are things going? How's the screen progressing? They send data, I send data, et cetera. And then at the end, of course, the hope is that we leave you with lots of data and hits, and you can go off and study the biology that triggered you to do the screen in the first place. Or to put it in IKEA cartoon terminology, don't try to build it yourself. Have a friend, and then you will have a smiley face. And I should point out that over the last just 5 years, platform scientists have co-authored 114 papers with other BroadEs in the community. So we think this is a model that scales pretty well. So we always have bandwidth to help you take on your screening project because of the way we've set up this model. Okay, so as I mentioned, we're going to have a bunch of different talks through the day. The next speaker is going to be Federica Piccioni, one of the screening scientists in the group. Federica.
Yes, so today I'm going to try to walk you through all the options that you have when you want to plan genetic screens and give you some examples. So how can you use the genetic screens to tackle your scientific questions? So you can use genetic screen to answer a question about a gene function, like which gene regulate expression of a reporter gene. You can use a genetic screen to answer a question about cell function, which genes are important for cell survival, or to identify potential drug mechanism or targets, like which genes enhance the drug effect. The choice that you have when you plan for a genetic screen are two. You can do pooled or arrayed. And in the pooled screen, each cell in mixture gets a different perturbation. Pooled screen is a very cost-effective way to query genome-wide libraries. And also, they allow you to do long-time course assays. On the other end, array screens are done in multi-well plates. Usually they require robotics, but they have the simplicity that every perturbation is in one well. They allow you to do robust interrogation of each gene and also very specific readout, like high content screening on high-throughput screening and gene expression profile. So let's focus on the specific steps of every screen. When you, in the pool screening, the library are made as a pool, and pool as a virus. They are lentivirus barcoded library, and they are infected in bulk in the cells at the low MOI. The cells are grown usually in very big flasks, while in the array libraries can be plasmid, viral, or oligo libraries. They are made in multi-well plates. And the cells can be either transduced or transfected, again, in multi-well plates. Here I mentioned just the Cas9 cells, so you can make Cas9 cells up front if you plan to do a CRISPR screen with the shRNA library. Again, in the pool screen, you have cells now growing in big vessels, and you need to select them because most of them are not infected. And then you apply a selective pressure that allow you to enrich or deplete your hit phenotype. At the end of the screen, you collect the cells, you extract the gDNA, you extract the barcode that you integrated in the genome, and then you PCR out, you amplify the barcode, and you submit for the next generation sequencing. In the end, what you're looking for are the guides, the perturbations that are enriched or depleted over time. On the other hand, in the RAID screen, you can either or not apply the selection, depends on the MOI. Usually it's very high MOI, so you don't need that. Then you can apply perturbation, it can be a drug treatment or anything else. And then a few days later, you acquire your phenotype, and in the analysis allow you to rank the phenotype of every perturbation. The cell number is a very important consideration in pool screens. We're going to focus now on pool screen. The RAID screen will be covered later by Brianna. Because to properly sample the library, we recommend that each perturbation is present at least 500 cells. And the majority of the cells need to be infected with only one perturbation. And when we talk about 500 cells, we talk about an average. So it is a distribution. So some of them will be actually low represented. And just to give you an idea about the numbers, so if you want to do a genome-wide screen, a genome-wide library like CRISPR's, you need 40 million of cells surviving after the selection, which means that you need to start around 100 million of cells and the multiple replicates. We are talking about 300 or 500 million of cells to do this kind of genome-wide library and screen. However, if you don't have this number, so you can actually think about smaller gene sets. And we have pre-made library, like a kinome of epigenetic, or you can actually build your own library, your custom library with the genes that interest your biology. It's important you infect a sufficient number of cells and you maintain the library representation throughout the experiment. This will be explained later by Yenare. So in the bottom line, really, in the pool screen, you really need to use enough cells to give statistical significance to your data. When you think about cell-based genetic screens, you need to take into account three components. You need to have a good cell model, a model that actually recapitulate the biology that you want to study. You need to have an assay, you need to have a readout of the cell state. And also, you have to think about the perturbation that you want to use. You can use loss of function, you can use gain of function, you can use genome-wide, you can use small set. So all this part will be covered by Kendall later. The most important thing is to understand that all these three things are interconnected. So these need to be planned carefully up front. When we discussed in the first meeting, in the intake meeting, that's all the questions that we're going to ask, and we're going to brainstorm. So what is a good model? So you need to have a cell line or a tissue that model well the biology that you want to study. But it also has to be feasible with the assay that you want to run. So you can have any primary culture or in vivo mouse or immortalized cells or iPS. But at the end, and this choice should be driven by the biological relevance. But at the end, everything is a compromise between the biological relevance and the feasibility of your screen. There are many assay types you can do in pool screens. The most typical that you probably have heard of is the viability, where the winners grow and the losers die. And it also can be done in vivo. You can sort by fluorescence. You can have a GFP reporter in your cells, but you can also fix and stain the cells and look at the endogenous gene. And the assays that are available, you can study migration using Boyden chambers, where the winners walk and the losers are left behind. You can use GILA, the growing low attachment assays, again, where the winners grow and the losers die. But in general, you can use any type of assay that physically separates the hit cells from the other. There are two flavors pool screen, you probably know. This is a positive selection, when you're actually interested in a very small set of winning perturbations. For example, if you want to find genes that overcome sensitivity to a drug. Or the negative selection, where the majority of the library will be still present in the end, and you're interested in those that actually disappear. You want to, for example, find perturbations that are lethal to the cells. Or, for example, in cancer, the genes that kill the cells, like in Achilles. I don't know if you have heard, which is a viability-based dropout screen. In this type of screen, the cells, shRNA, if we use the shRNA library shRNA screen, parental cells, if you use CRISPR screen, Cas9 expressing cells, are infected with the genome-wide pool screens. the cells after infection are selected, they are allowed to be passage for 2 or 3 weeks, because in this type of screen, you need the loser to dilute out. You need time. And after 2 or 3 weeks, you harvest the genomic DNA, as I mentioned before. You amplify the guides or the hairpin by PCR. You submit for Illumina sequencing. And at the end, you're looking for the guides that are depleted in the selected population compared to the original on an earlier time point. Another type of viability assay that you can do in pool screen is a rescue screen, where you actually have a phenotype. In this case, is a drug sensitivity, saline, which is very sensitive. You introduce any perturbation that you want, and you're looking for genes that overcome this sensitivity, basically they confer resistance. In this example, Stephen Whittaker, who worked in a Galway lab at that point, he used a cell line, a BRAF mutant melanoma cell line that is sensitive to the BRAF inhibitor like PLX4720. He infected these cells with an shRNA genome-wide library. He selected, and after selection, he split the cells in 2 arms, an untreated arm and a drug-treated arm, and let the cells go for a couple of weeks. And he was looking for a guide, in this case of the shRNAs, perturbations that were conferring resistance to the drug. They were enriched over time. And those that are enriched gene, the top 5 enriched gene, and the top hit here is NF1. And when he went to validation, he showed that the shRNA targeting NF1 is able to suppress the protein by Western blot. And when he did a colony formation assay, he showed that the cells grow in any condition in absence of the drug. But when he treated with the drug, only the cells in the SHA treated with the shRNA targeting NF1, the cells were growing, suggesting that the NF1 is important to conferring drug resistance in this melanoma cell line. Now I want to move away from the viability assays and talk a little bit about the FACS-based screen you can do in a pool screen. And in this screen, the aim was to find new therapeutic target for HIV. So in HIV, the proteins interact with the host cells protein for entry into the cells, for the replication, and for the transmission. So everything starts when the envelope protein engages CD4 receptor and the CCR5 co-receptor, the 2 membranes, so there's a fusion of the 2 membranes, and the virus entry those cells. So it's very important for a therapeutic reason to find the host protein, the targeted by HIV. And here I describe the cell model and the assay that was developed to study HIV infection. So they use a T cell line expressing CD4 and CCR5 that was encoding for GFP driven by the HIV LTR, which means that when you infect the cells with HIV, the cells turn green. So you increase the GFP expression, as you can see here. So you actually can use, and you can also monitor these using FACS. And so GFP actually can use as a reporter for HIV infection. I want to just notice that this is a very low background as an assay, so this is a very good model. So Ryan Park from Nira Cohen lab used this cell model to do a genome-wide CRISPR screen. He used these cells. He engineered expressing Cas9, and then introduced a genome-wide guide-only library in these cells. He selected them, and at the end, after selection, he split them in 2 arms. One was infected multiple times, challenged with HIV, and the other one was untreated, left untreated. At the end, he had cells surviving, and in this population, there were either all green cells or not GFP expressing cells. And the green cells were the cells that increase HIV infection, while the less GFP expressive cells were actually expressing the guide that prevent the HIV infection. And this is the histogram, the flow cytometry histogram, just to show you what is the separation of the assay. And when you infect these cells with HIV, a specific strain of HIV, the cells turn green. But if you infect them with the guide, for example, CCR5 guide, which should be prevented HIV, the cells express much less GFP. Those are the results of his screen. The top 5 hits enriched in this population. And nicely, it was nice to see that CD4 and CCR5 were among the two, there were 2 among the 5 hits. And he was able actually to find also other targets that prevent HIV. Another interesting assay you can do genetic screen for cellular transformation is GILA, Grow in Low Attachment Assay. I don't know if you study cancer, you know what is a soft agar. It's a very low throughput, robust, but time-consuming assay to study in vitro cellular transformation. So Asaf Rotem developed this assay, the GILA, in order to replace the soft agar, specifically for high throughput. So you can actually run multiple genetic screen or drug screen with this assay. And the GILA assay utilizes the unique ability to transform cells to grow in anchorage-independent conditions. So for example, when you do soft agar, the colonies that you see in transformer line look like this. When you do GILA in low attachment, the cells grow in aggregate like this, but phenotype is pretty similar. So Asaf actually did a North screen to study genes that specifically increase transformation using GILA. He took the cells, he see them in 2 different conditions. One is the low attachment, the GILA, and the other one in high attachment. The following day, he is infected with the North genome-wide North library. And a few days later, cells in GILA were in suspension, cells in the high attachment were attached. He collected the cells, and he looked for the abundance of each perturbation in each sample and compared the low versus high attachment. So here's the comparison between the high attachment versus the low attachment. And those are the genes that are highly represented in GILA, and that should be oncogenes because it increase the transformation. And in fact, he found that HRAS, KRAS, EGFR are among the top hits, which is very, very nice to see. Other hits he found in his screen are kinases, receptor, and signaling pathways linked to cancer. Another assay is cell migration using Boyden chambers. So in Boyden chambers, you basically seed the cells in a chamber that is suspended above a well. And the cells, invasive cells, they are able to migrate, are actually able to move through these membranes. So using this technique, in these screens, so the cells, which was a very low background cells, were non-migratory cells like MSF10A, this is the breast cancer cells, were infected with the genome-wide HRNA library. And they were infected, selected, and then put in a lot of Boyden chambers, and then they allowed the cells to migrate. At the end, they selected the cells that migrate, the move on the bottom of the plates, and they were looking for which guide and were enriched, which guide allowed the migration of the cells. And this is representative migration phenotypes from shRNA that came out from the screen, and this is the background. As you can see, very few cells are able to migrate. This is a very weak phenotype, and this is a very strong phenotype where most of the cells migrate. And again, in these screens, they were able to find a gene, DLG5, which knocked down, increased the cell migration that they repeated in a low throughput, again in the Boyden chamber, and in another migration assay, like the Scratch assay.
For something this size, do you have any idea of how many chambers?
I think there were hundreds.
Yeah, I mean, they did it in 24 well-plate format. So, you know, fair amount of elbow grease, but, you know, you do it once, get the data, and you forget about it.
I should point out that we no longer use microarray analysis.
Okay, this is an old screen, but
Published in 2010, no microarrays anymore.
sorry. And then, sorry. As probably you know, you can also do Perturb-seq using pool screen, where you combine CRISPR technology with a single-cell RNA-seq as a readout. You infect the cells with a barcoded CRISPR library. The mRNA of the individual cells then is extracted with a uniquely barcoded primers. And then the single-cell RNA sequencing is able to reveal both the CRISPR-targeted genes and the resulting transcriptional profile of the single cells. So the bottom line that you barcode both the individual perturbation and the cells affected so that the sequencing can identify both. The last screen, this is the last, I promise, is an in vivo screen that Robert Mancuso in Nick Haining lab did to find new cancer immunotherapy targets. He took the B16 cells, he engineered with Cas9, introduced a library. So this is an in vivo study. Again, you can't do it genome-wide. So he made his own library of 10,000 guides, 2,500 genes, 4 guides each. He infected the library, he selected, and then he injected the cells in different mice. Probably, how many mice?
10 to 20 per group.
Okay, so this is the number. So he introduced the selected population in T cell deficient mice, in wild-type mice treated with the GVAX, or in wild-type mice treated either with a combination of the GVAX with the antibody against PD-1. And this is the volume of the tumor. So the tumor grows very nicely in the T cell deficient mice. They are smaller when treated in GVAX, and decrease in volume again when they are treated with a combination. And they collected the tumor here, strategy DNA.
Just wondering, for the in vivo tumor genome-wide, have you found some issues with the stochasticity of the cells that actually make the tumor in terms of maintaining representation?
I haven't done that.
Yeah, so I mean, that is exactly the issue with in vivo screens, is you don't know a priori. You know you put 1 million cells into the mouse, you have no idea how many contribute to the tumor, which is why step one of any screen like this is to empirically determine how many cells are contributing to your tumor up front. And when the Haining lab did this, they realized, well, wait a minute, if we want to do a genome-wide screen, we'd need 2,000 mice. So that is why they designed a much smaller library, only targeting about 2,000 genes or so. And then they realized, like, well, okay, if we combine enough mice together, then we maintain good representation. Exactly.
This is not genome-wide.
Oh, sorry.
This is a very small analysis in guides.
So I have another question regarding that. So would it make sense then for these screens to run in parallel the same screen with cells that don't express the Cas9?
For the statistic effect, to see which ones.
Because if you're running in parallel cells that don't express Cas9, whatever you get in the end, in that group, will tell you how many cells you ended up with.
Right, so this is exactly the strategy that one uses. When you want to do an in vivo screen, to get at this question of representation, you need to put in some library that is not doing anything. Whether it's a guide RNA library that Cas9, whether it's a library of random barcodes, whatever it is. But you put in a library and then you ask the question at the end, well, how many barcodes do I get back out? And that determines sort of the carrying capacity of your in vivo model. That is absolutely part of the assay dev one needs to do. And when you do that in advance to determine the scale of your screen.
Yeah, that's what I'm thinking of, though, even though you have some idea in it. So to save time, Because
No,
the whole,
no, you'll waste time. You need to determine that number up front.
so if you have a low number, say you're going with 2,000 guides, right?
Right, but 2,000 could require 5,000 mice based on your model. And so if you do it in parallel, I mean, it's possible.
I'm trying to think, what's the event? Because when you're going to run the screen, you're going to have a lot of non-targeting guides, right? So is it better to have a lot of non-targeting guides than to have the Cas9 non-expressing cells, or both?
Well, there are multiple ways of determining noise in a screen, for sure. And we can talk about that offline. But in general, step one of any model system is determine how many cells can I effectively study. And that is by far best done in advance, not guessing that, well, I hope if I do my screen with 20 mice and I have a library of this size, that it will work out okay. You determine in advance empirically with barcodes.
Okay, so just a moment. So this is the distribution of the data, the entire library, and in red you can see each guide has PDL1, and you can see the PDL1 here is depleted in the GVAX mice compared to the T-cell deficient mice, suggesting that PD1, the loss of function PD1 can actually increase the sensitivity of cancer cells to immunotherapy. And this is all the results of the screen, suggesting that he identified other targets, they increase the efficacy of immunotherapy, suggesting this screen worked very well. And the last, I just want to say that only a cell autonomous effect will enrich or deplete cells carrying the perturbation that causes it. So you can do a pool screen in genome-wide library, equaling the genome, do a long-time course assay, but specifically you are looking at the phenotype of the perturbed cell. And non-cell autonomous effect can be missed in these screens. And now I move on with Brianna, who's going to talk about the array screening.
Hopefully people can hear me. So I'm Brianna, and I'm going to be talking about array screening. So I'm going to kind of go more into how you would want to do an assay development using a 384 well plate. So just a quick follow-up with the pooled screening. If you want to do a follow-up screen, array screening is a good way to do that. So there are different types of arrayed assays. So first, starting with a very simple assay would be cell viability. This you can do using cell titer glow, Alamar Blue, or just imaging. There's also reporters using luciferase and GFP, gene expression using QRT-PCR, RNA-seq, or L1000. There are also more specific arrayed assays such as high content imaging and low content imaging. So a simple assay such as cell viability is going to be better for high throughput. However, more complex assays such as high content imaging and low content are going to be more specific, but they're going to come at a higher cost. So these are all the arrayed assays you can choose from. So depending on what kind of screen you want to do, you have multiple assays to choose from. So in order to get an effective lentiviral transduction, there are many key variables that you need to factor in and optimize in order to get an effective transduction during your screen. So you're going to want to start most likely first with cell density. So cell density is really important because obviously it can vary from cell type to cell type. So you want to make sure that when you seed your cells, you want to seed them at the right density where when you infect them, they're going to have a good infection efficiency. But you also don't want your cells to be too confluent by the end of your screen when you want to read out. So another consideration is polybrine. So you want to look into the concentration of your polybrine before you infect the cells. You want to make sure that the concentration you're adding isn't too toxic to the cells even before you start your infection. For selection, you're going to want to choose a selection marker that is appropriate to your library. For example, drugs such as puromycin and blastocidin are used a lot. And with these selection drugs, you want to make sure the concentration in the dose that you're adding is correct. So you want to make sure you're adding enough puro that you're killing the cells that are non-infected so that you're not getting too much background. For incubation time, you want to really look into how long you actually want to run your screen, and also go into other factors of how many media changes you want to do, how much you want to replenish your media to help with your cells. One of the really big things you want to look into is your virus concentration. So how much virus are you adding into your cells? This can even vary among your library plate. So some wells might be easier to infect than others, so you want to make sure you get a virus concentration that's going to actually infect well those low titer wells. So I will go into explanation of how you're actually going to assay and optimize this virus concentration in a bit, as well as you need to take into consideration plate effect and edge effect. So I will explain this a little bit later, but there can be some circumstances where you see some evaporation in your plates during your incubation time, and this could affect your results in the end, but we have ways to reduce this effect. So by looking at all these key variables, we want to get a high transduction rate. And the way we do this is we do an assay that is a 3-step process. So for step 1, we really just look into optimizing the cell density, the polybrane concentration, and the dose of your selection drug. Once these 3 key variables are optimized, we then move into step 2, where we're going to estimate the virus dose using control viruses such as GFP, LAC-Z, RFP, and BFP. So these are just a few. There are many other control viruses to choose from. And once we kind of get a good gauge of what virus dose to use for your cell type, we then move into step 3, where we optimize the virus dose with a plate of control viruses that are actually going to be used in your screening library. So to roll back to step 1, this is a 384 well plate, and what I'm trying to show here is that your 3 key variables that you want to optimize, such as cell density, polybrane, and drug, can actually all be done in just one plate. So the way we would normally go about this is over here, we'd have 4 to 8 seeding densities. So starting at the top of the plate, we'd start with the lower seeding density and increase as we go down the plate. We then will break up the plate into 3 sections and have 3 different polybrane concentrations. You obviously want to control here, so you don't want any polybrane to see if there are any toxicity effects in the other doses. And then over up on the top here, obviously in these 3 quadrants, we're going to have a selection of puro and blast, and we can have the drug dose increase in these 3 sections. So what's really nice about this is you can just use one plate to optimize all 3 of these variables at once. So after those 3 variables are optimized, we then move into, as I mentioned, the virus dose using a few control viruses. So what I'm showing over here is a 384 well plate again, and what's in the box in red is basically I'm highlighting one quadrant of the plate. And what you can do here is you can infect with 2 virus volumes. So the one column is going to get 1 microliter and the other column will get 2. But one row will get no puro and then the other row will get plus puro. This is how we calculate infection efficiency, by using a very simple calculation of puro divided by no puro. From here, you can then generate a graph of your data to look at infection efficiency. And what you're really looking for is to see out of which virus concentrations you use, which actually gives you the best infection efficiency for your whole entire plate. So you want to look more up in this area here to see, and then you can see that 0.5 microliters has a pretty good infection efficiency. So how you would do this readout for infection efficiency is you would use cell titer glow. It's a very simple assay. It doesn't take very long to read at all, and we have the machine to actually read out your plates. So after we have estimated a virus dose, we then optimize the virus dose with the controlled virus plate, which is the same test with the screening library. So we kind of have a good idea of what virus concentration to use, but not the actual one for your screen itself. So we usually will infect, again, in a 384 well plate, usually 2 to 4 virus concentrations. And then, once again, test infection efficiency to see which virus concentration is best. Yes?
Just to the previous slide, have you found any issues or discrepancies when using cell titer glow as an infection efficiency rather than cell number or a final readout?
Depending on if you let your plate, you have to let the plate sit out to warm. You need to make sure that the cell titer glow is at a warm temperature. So basically, we have everything set up so that when you repeat the assay, you shouldn't have any problems.
So it's not so much the actual technical assay, but the readout of the assay being ATP rather than cell viability.
No, we have seen very consistent results. And we tested many cell lines over the years. I can recall any specific effect.
I mean, you raise a good point. Like, in the high-throughput screening world, if you're doing small molecule screening looking at viability, there are many papers showing that, like, well, if you read it out by Alomar Blue versus by cell titer glow, different small molecules score. That is absolutely true because they're looking at different aspects of cell physiology. Whereas here, you're just looking at within the same cell type, plus puro versus no puro. So it's at least internally controlled that way. And when we've done assays like this with Alomar Blue, you get basically the same result because it's internally controlled. But in general, for screening, that's exactly right. ATP readout, cell titer glow, is not the same as other readouts for cell viability. They're all surrogates of something. Yeah.
So for reading out again for this, we still use cell titer glow, and we have suggested infection volumes for either high titer virus or low titer virus. So for high titer virus, we suggest anywhere between 0.5 microliters to 2 microliters of virus per well, and for low titer virus, we use between 4 to 12 microliters of virus. So things that can go wrong during your screen. So a lot of people will design library plates where their whole plates, so for example, all of these red wells are wells that contain virus. So that when you go do your infection, all of your whole plate will have all infected cells. You really need a few non-infected cells in order to test infection efficiency, and also make sure you're not getting any high background noise with your puro dose. So what I'm showing over here is the same exact plate. So all the red wells still are wells that will contain virus, but I have left 3 wells in white, which are essentially empty wells. So there will be wells that still contain media, but they actually don't have any virus. So when you're doing your infection, you will have 1 to 3 non-infected cells. This is also really important for plate orientation. So you could be rushing through the day and you could accidentally infect with your plate completely backwards, and you could go through your whole data screening and get completely wrong results because you accidentally flipped the plate. Well, with this, having some empty wells, you should be able to recognize and see that you actually switched the plate. So hopefully that won't happen, but it is a really good QC point in your screen. So another thing that can go wrong that I mentioned before is edge effects. So this 384 well plate here shows actually a really bad edge effect. So the whole outside is red, in the middle is green, which is actually what you would want to see throughout this whole entire plate. So a lot of times this is due to evaporation during the incubation stage of your screen. So the ways that we can reduce this is by optimizing the seeding density of your cells to make sure that your cells on the outside of the wells don't get affected. And normally we would do this by making sure that we'd maybe replenish your media more often, which then you would need to take into consideration when seeding, because if you're replenishing the media, your cells might be growing faster during your screen. And then for incubation, we normally recommend putting water plates on top and bottom. I know this seems kind of tedious, but it will really help prevent evaporation of your plates while they sit in the incubator. And for media conditions, like I had mentioned, you would do replenishing of the media during your screen itself. Or also, we maybe will just add 50 microliters more media to your wells so that if some evaporation does occur, it shouldn't have any effect on your screen, and we shouldn't be able to see any of this. It should all look more of a uniform color of green. So once we have optimized and done all the assay development, we then go into an arrayed screening workflow. So if you start here on the left, you will seed the cells. If you need my help, I'm there to help you use this robot to help seed. We then take the library plates and infect your cells with them. We will then do a drug selection on your cell plates, and then we will perform a viability assay for QC. So this is still usually cell titer glow, so we'll always have a puro and no puro plate as a control, no matter what assay you're doing, because we just want to make sure that the infection actually worked before you spend your time going through all of your other data analysis. You will then measure your phenotype with the assay that you have decided on, and then analyze the results. So arrayed screening can be a lot of work and a lot of time in optimizing. So how can we help with your screen? First, we design your library with you, containing both positive and negative controls, which are very important and you don't want to forget. We then generate the lentiviral library for you. So essentially, I will be making your 96 well plates containing all of your virus. I can also rearray them into 384 plates, however you want, basically. We then help with designing and optimizing the screen, doing the assay development using our robotics in our BL2 Plus lab by using this 3-step protocol that I had went over. And then we execute the screen with you. So basically, you prepare all your medias for me, you hand me your cell plates, and basically, I run the programs that are needed for your screen, and you don't have to worry anything about the robotics. Basically, I hand you back your cell plates, and you're good to go. Put them back in the incubator, and then you don't have to worry about anything else. And then once you have your data, we can help you analyze, normalize, and interpret your data, and then help you if whether or not you want to go and do another screen. So basically, we really help guide you through this whole entire process, which is very helpful for this big arrayed screening. And these are just 3 of the robotics that we have in our lab upstairs. So with that, if no one has any, you have a question?
It's very robust, you can work, even with 2.
Okay.
But for nausea isolation, it's really good.
Yes. What's the typical confluence that you look for at time of infection?
What density for time of infection?
Like, what's the ideal confluence? If
It depends on how the cells grow. If there are fibroid cells, you can have 250 cells per well. But if they're very slow growing, it's not so fast. Okay, about 80 per well.
I'm sorry, I can't really hear you.
So,
250 to 1,000.
So it's going to be very condition specific. So some cells, if you treat them with polygreen at a low density, they'll die. Whereas if you see them at a high density, they might be fine for infection, but then they'll hit confluence 36 hours in your assay. What if your assay is a 5-day assay? So there is no one right answer for what is the right confluence for your cells. That's why you have to go through and optimize all these things together, because there is no one specific answer for it. It's very, very assay specific.
Yeah, I'm just thinking, like, so I go and I do that optimization of that plate, and then how do I decide which one?
Oh, you do titration. You put many CD density in the plate. At the end, if you have a 3 days assay, if you have a 6 days assay, then we look at the confluence.
Just as one example, so if you seed it too high a density and then you go to select with puromycin, not all the cells will die if they're too high density, because drug selection is often density dependent. So that would be bad, because then you're not removing infected cells. Likewise, if it's too low, then the polygreen might kill them. So it's really a question of what is your endpoint and work backwards from that to find the right conditions.
Okay. And now Kendall will talk about perturbations. Hi, I'm Kendall, and I'm going to talk about the different
applications that one can do with CRISPR. So as Federica discussed, to have a good screen, it's really important you have a good model, a good assay, and then a good perturbation. So that's what I'm going to focus on. Perturbations can be divided into two major categories. We have loss of function perturbations, your classic CRISPR knockout, CRISPRi, and then your shRNA. You can also have gain of function perturbations, which would be CRISPRa and ORF screening. So it's not a question of what type of screen you should do, but which first. If you have a good model, we recommend you do many screens. With each perturbation, you'll, learn a new thing every time. So I'm going to jump right into CRISPR knockout screening. To successfully have a good perturbation, it's really crucial that you have a good guide RNA. And we see a lot of variability in guide RNA efficacy. So here we have the gene NF1, and on the x-axis, amino acid position, and then on the y, we're looking at log fold change. Because this is a positive selection screen, we're looking for enrichment, so a positive log fold change. And as you can see, we have guide RNAs tiled across this entire gene. Each point is a different guide RNA. And there's not one specific site on the gene where you see guides working better than others. And you see that some guides work and some really don't. So it's really important that we design these good guides. So what we then set out to do is create rules to look at nucleotide preference. And we can use these sequence features to design better guide RNAs, which lets us design better libraries. And as GPP exists as a platform, we've continued to create better rule sets and therefore better libraries. So we first collaborated to make Gecko v1, which then came Gecko v2. We were then able to create rule set 1, which led us to design our human and mouse libraries, Avon and Asiago. Then created rule set 2. We now most recently have Brunello and Brie. For any Epicureans in the room, we like to name our human libraries after wine and our mouse libraries after cheese. So that's kind of a good way to navigate our platform if you're a little confused on the names. That too. So what you then do when you have your data set, you want to look at how do these libraries compare and what's their performance like. So we use these predefined gene sets for essential and non-essential genes. And when you rank all your guide RNAs, you can see that of your non-targeting guide RNAs, those are the least depleted in your data set, whereas your core essential guide RNAs would be among your most depleted. And this is a really great metric to compare library performance because you're not collapsing guides to the gene level. You're taking every guide RNA into account, which allows you to compare performance of libraries of different sizes. So we then take these predefined gene sets and we will then plot the percent rank of guide RNAs against the cumulative percentage of the guide RNAs in that set. And we then will find the area under the curve. So here you see the essential genes have an area under the curve of 0.8, whereas your non-targetings would have an area under the curve of 0.16. And we then plot these area under the curves to kind of see how libraries can compare against each other. And as library design improves, you would expect to see increased separation between your core essential and your non-essential genes. And we're going to refer to this as delta AUC. So we then plot this delta AUC to see how libraries stack up. And as you can see, all the points in red are the GPP, genome-wide CRISPR knockout libraries. And you see that Brunello, our newest library, is doing the best yet with the highest delta AUC. And by this metric, you also see that Brunello compares very favorably to other published genome-wide libraries out there. So in order to execute a CRISPR screen, there's two components. You need your Cas9 and you need your guide RNA. And there's two different ways you can approach it with CRISPR knockout. We have our split vector or our lenti-guide approach, where you would do an infection to establish Cas9 in a cell line. You would then do a second infection, which would deliver your guide RNA in a separate vector. We also have a all-in-one or lenti-CRISPR V2 approach, where both your guide RNA and your Cas9 are being delivered in the same vector. And there's different cost benefits for doing both those in terms of execution, which Zora will cover later. But when we look at performance, you really see no difference in performance if you do one way or the other. So there's many components of the CRISPR system that one can modify, the idea being that you want to increase your on-target activity while mitigating any off-target effects, which can be problematic when you're screening with CRISPR knockout. So people have proposed modifying the Cas9. Both eCas9 and Cas9 high fidelity are out there, and they're based off this energy hypothesis that perhaps any off-target activity can be minimized, while your on-target activity can be maintained by weakening these interactions between Cas9 and your target DNA. So we have eCas9 targeting with modifications along the non-targeting groove, and Cas9 high fidelity with these modifications along this Cas9 DNA contact site. People have also proposed modifying your tracer RNA to try and achieve the same thing. So we created tracer v2, where we did a similar nucleotide swap to here, and we also had a 5 nucleotide stem loop extension. We then want to see, okay, how do these modifications affect screening? So we created a tiling library with guides targeting EF2, which is a gene that's a known cell viability gene, as well as guides targeting HPRT1, which is a gene that's known to confer 6-guanine resistance when knocked out in cells. We clone these into all-in-one vectors with your Cas9 high fidelity, your eCas9, and then the wild type. And we also had the wild type Cas9, but with the modified tracer. And we then did both a dropout and a positive selection screen. In dropout, you would expect cells with EEF2 knockouts to deplete. In your 6-guanine arm of your screen, you would expect cells with HPRT1 knocked out to enrich. And we then looked at these results to see how these modified elements do in a screening capacity. So here is EF2 knockout cell viability data, and you're looking for depletion here. So as you can see, on the x-axis, we've plotted log 2-fold change of the wild type Cas9, and you see really good depletion from wild type. Whereas when you look at the y-axis for both eCas9 and Cas9 high fidelity, you see not a lot of activity, and therefore they do not do as well in retaining their on-target effects as the wild type does. We also want to look at tracer v2. So on this far panel, we look at perfect matches, which would indicate your on-target activity, and you see significantly more depletion with tracer v2. That being said, you can look at mismatch positions, and we also see more depletion from tracer v2, indicating that it has both increased on and off-target activity. So what do we recommend when you're screening? Do not use eCas9 or Cas9 high fidelity in a screening capacity. They seem to not have as good on-target activity as your wild type. However, if you wanted to use the modified tracer versus the wild type, the effects are neither helpful nor harmful. To confirm this, we did a genome-wide screen with Brunello, with both the wild type and the modified tracer, and once again, we're looking at this area under the curve metric to look at performance. And as you can see, both of them perform comparably. So I want to go into a different application of CRISPR, CRISPR interference. And for both CRISPRi and CRISPRa, we use this catalytically dead Cas9, which will not cut your DNA. However, for CRISPRi, there's been a lot of tech dev out there, and we found, or other scientists have found that dCas9, when fused to a crab domain, has significantly better gene silencing than just dCas9 alone. So when you're designing guides for CRISPRa and CRISPRi, as I mentioned, with gene knockout, your sequence is so important. However, when you're looking at gene expression, there needs to be a balance between location and your sequence. So we are looking at CRISPRi location preference, and these are 3 different databases that annotate your TSS site. And it's really important that you have the best annotation possible so that you can have the best guide design possible. And as you can see, Phantom performs the other 2 existing databases, and that's because it's based on actual data, whereas the other 2 are models. And so we see this really high peak in activity right here. So that's the site that we decided to target when we designed guides for CRISPRi. There are 2 currently existing CRISPRi libraries out there from Weizmann. We also have developed human and mouse CRISPRi libraries, Dolcetto and Dolomiti, and each is divided into 2 different sets. Set A is your top 3 guide RNAs, and set B would be your next 3 guide RNAs. In terms of how to execute a CRISPRi screen, it's very similar to the split vector lenti-guide approach that you might use for knockout, where you would establish cell lines expressing dCas9 crab, and you would then infect your guide in a separate vector. And just worth noting, our CRISPRi library comes with the modified tracer element because these on-target activity, our on-target is really important for CRISPRi, and off-target is less of a concern. You're not having a permanent mutation. So how does our library stack up? When we look at the same delta AUC metric that I described earlier, we see that Dolcetto is significantly doing better by this metric for CRISPRi libraries, but was also a nice validation is you see that it's also comparable to CRISPR knockout, which was cool to see. There's certain considerations one must make when you're considering loss of function screens. Obviously, CRISPR knockout, it's a double-stranded break, so it's a permanent mutation, whereas CRISPRi can be turned off. Both our CRISPR knockout and CRISPR libraries perform comparably, and your CRISPRi guide design can be a little more difficult. You have fewer choices because location is so important. However, there's fewer chances for any off-target effects. There's a caveat with CRISPRi, which I'll go into in a bit, about bidirectional promoters. However, with CRISPR knockout, you get this unwanted cutting effect, which in chromosomes, you can get these copy number amplified regions where you have genes scoring as essential, which is a source of false positives due to this amplification of cutting. However, this cutting effect disappears with CRISPRi. You can see when you compare your non-essentials with your non-targeting, they're having similar AUCs, which indicates this loss of cutting effect. You do have an issue maybe of bidirectional promoters with CRISPRi. So here is for 2 genes, this is their TSS site, and you see promoter activity on either side. So when the dCas9 goes to the target, you see silencing of both genes with CRISPRi, whereas with CRISPR knockout, only one is essential.
For sensitivity screens, like looking for dropout on drug treatment, have you found that the CRISPRi has been able to be a platform to interrogate gene essential genes, whereas you wouldn't be able to look at these in standard CRISPR knockout situations? Or do you still see a robust dropping out of these essential genes?
I think you still see it in both.
Yeah, I've been trying to get people to do that experiment, so if someone wants to, please let me know. No, I mean, like, so when we look at these core essential gene sets, we see CRISPRi and CRISPR knockout perform comparably. You know, they're both good at depleting ribosomals and proteasomals, but that's kind of unsurprising. You can imagine that even a 50% reduction in ribosomal genes is enough to cause a viability effect. What we don't know is for very stringent screens. For example, you know, the one Federico was talking about earlier about how does HIV get into a cell. You can certainly imagine that if you still have 10% of a receptor around, that's plenty for HIV to still get in. So what we haven't done yet, and we wanted to sort of do with the community, is get a sense for, in the same screen, when it's a really strong selection phenotype, does CRISPRi perform comparably to CRISPR knockout or not? That we just don't know yet, but it would be certainly very interesting to compare.
We're going to do it, Jim.
Nancy's going to do it. Great. So come back to next year's BroadE, and Nancy will be presenting on her data.
So now we're going to move into
CRISPRa screening, which would be a gain-of-function perturbation. So there's quite a lot of evolution that went into the CRISPRa system. Originally, there was just dCas9 tethered to VP64, and they did not see a lot of amplification with that. And then the SunTag system evolved, and now the assembled SAM complex is Also, you still infect your cells with the dCas9 tethered to the VP64. However, you have these additional PP7 stem loops, which recruit additional activation domains, the P65 HSF domain. So similar to CRISPRi, location, location, location for guide design. Phantom is obviously giving the best annotation here, and we see this nice peak in activity here, so that's what we targeted for our CRISPRa guide design. Similar to CRISPRi, we developed genome-wide human and mouse CRISPRa libraries. We have Calabrese and Caprano, divided into 2 sets with the same idea, your top 3 guides and your next 3 guides. Also, similar system to CRISPRi, you're gonna infect cells with your dCas9 VP64, establish that line, and then your guide RNA comes in a separate vector. However, this one has the additional transcriptin, activators, the PP7, P65 HSF. So we repeated the same vemurafenib resistance screen that Konerman did, and when we compared Calabrese to the SAM library, you see at certain p-value cutoffs, we have more genes scoring, which was a nice validation. And so I just want to reiterate, if you have a good model, you want to do as many types of perturbations as you can. You'll learn new things every time. We do have activity assays available for each type of screening, so you can confirm your Cas9 or your dCas9 presence in your cells before you proceed with a screen. And I want to move into kind of a different idea, which is this combinatorial CRISPR screening. And this is the idea of knocking out 2 genes at once, kind of based off the whole concept of synthetic lethality that exists in cancer biology. So people have been trying to find good ways to knock out 2 genes at once for a while now. There's this issue of recombination, but also competitive association. We developed a 2-Cas9 system using S. pyogenes and S. aureus Cas9. And because it's 2 different Cas9s, you get no recombination, but you also get no crosstalk in your system. And we found it to be very effective for double knockout. So we originally tested it with a guide targeting GFP and one targeting CD81, and we see consistent double knockout achieved with these orthologous Cas9s. We then tried a pooled screening approach, which, similar to standard, you have a guide for each Cas9 with M plus N oligos, and when you clone that, you get M times N combinations. Because you're looking at double knockout, you're now kind of working in a matrix space. And how you execute this screening is you would, similar to your lenti-guide approach, you establish cell lines expressing the SP Cas9, and you would then deliver both guide RNAs plus a vector with your Assay Cas9. We call it Big Poppy because the combinatorial space is large and it's paired aureus and pyogenes for interactions. So one combination that scored high was the BCL2L1-MCL1 relationship, 2 anti-apoptotic genes. And when you look and you have your BCL2L1 paired with a control, and same with MCL1, you see little to no effect. However, when both BCL2L1 and MCL1 are knocked out, you see significant depletion. Similarly, when you switch the Cas9 position, you see the same effect. We also looked at another hit we got, the MAPK1, MAPK3 combination, same deal. When they're paired with a control, little to nothing, and when they're both knocked out together, you see depletion, and same when they're switched. So kind of to follow up on this BCL2L1, MCL1, we looked at a small molecule times small molecule matrix, and we had an MCL1 inhibitor, a BCL2L1 inhibitor. We see really nice synergy with this, and this is cell titer glow cell viability readout. And these are all the anti-apoptotic genes that we looked at in our library, and you see these very specific hits coming out of it, which is interesting. And just to kind of, there's exciting possibilities of the system. Double knockout is not the only combination. Each Cas9 is programmed independently. So you can have knockout with up, you can have up and down. There's a lot of different possibilities for the system, which is very cool. So just kind of a reminder, since we're working in combinatorics, it's easy for all the combinations you want to test to get ahead of you very quickly. There's only so much time to do TC in the day, so it's really important that if you're interested in looking at combinations, you're very specific with what you're picking and, you know, narrowing down what you want to study. So just to summarize, we have these great libraries available, both knockout, CRISPRa, CRISPRi, and our PPOPI vectors show promising opportunities for any types of combinatorial screening that people are interested in. And now we're going to hear from Zora more on how to actually execute these pooled screens.
All right, thanks, Kendall. So we've learned about all these cool different systems for perturbations, and now we're ready to start planning our screens. I'm going to be going over initial QC metrics, things you need to consider, and best practices before you get started on a screen. So an important thing to consider before you start any kind of screen is looking at publication requirements, specifically when it comes to cell sourcing. As an example, Nature has a reporting checklist for life sciences articles, and they ask a couple important questions. Specifically, they ask if you're using any cell lines that are commonly misidentified and to justify why you're using those cell lines. The two things I'll be focusing on is, have the cell lines been authenticated, and if so, by which method? And have they been tested for mycoplasma contamination? A few points on cell sourcing. Some trusted sources for cell lines would be ATCC, Sigma, DMC, which is based in Germany, and CCLE, which is internal. But regardless of where you're getting your cell lines from, it's incredibly important to authenticate on your side. I'm going to go over SNP fingerprinting. It's actually conducted over here by the genomics platform. Their protocol requires that you collect a cell pellet of about 5 million cells in PBS that you can store at -20. You would then isolate the gDNA from your pellet and plate into a barcoded 96-well plate that you can obtain from GP. You would then ship it to the loading dock, where it's taken to 320 Charles for processing and analysis. GP processes up to about 4,800 samples per week, and because of that, processing times are going to vary, so you can expect your results from anywhere between 3 weeks to 3 months based on their analysis queue, the availability of their reagents, and so on. An important point about fingerprinting is that you really want to make sure that primary or obscure cell lines that may not have an established SNP profile should be fingerprinted more than once and be compared to one another. So we recommend fingerprinting at the start of your screen and at the finish, and compare them both to make sure that they actually match each other. Another way to authenticate your cell lines is through short tandem repeats, or STR profiling. These repeats are specific to cell lines and are actually conducted internally through PRISM. You can contact Jennifer Roth for more details. The process is a little bit similar to SNPs, where you would pellet your cells, extract DNA, but you'd be running a PCR and comparing alleles to a reference database to see if your cell line matches. This can take as little as 1 week to process, and the results are pretty easy to interpret. Once you've authenticated, you want to do mycoplasma testing. Mycoplasma is incredibly ubiquitous. They can be present in anywhere from 10 to 85% of your cell lines, depending on the lab across the world. Signs and symptoms can include a change in media color, an excess of debris in your flask, changes in morphology and growth. But the thing about mycoplasma is that you can see no change at all in your cell lines and still have contamination. That said, it's still incredibly important to make sure all of your cell lines are mycoplasma free, because they can impact your screen quality and the results. Specifically, mycoplasma can cause drug resistance in positive samples and can change gene expression and epigenetic regulation. So a couple ways to test for mycoplasma are PCR and DAPI staining. To run a PCR, you would seed cells in a flask and keep them confluent for 3 days without changing the media. You would then collect a milliliter of the media and submit that for PCR. We use a mycodetection PCR kit from ATCC to amplify. And for DAPI staining, what you'd want to do is seed cells into a 6-well at 20% confluence and let them adhere for 4 hours. You'd want to fix the cells with 4% paraformaldehyde and stain with a dye. And then you'd want to visualize under a fluorescent microscope. The micronuclei will appear as halos around your cell nuclei. So here's what a couple PCRs look like for mycoplasma. You can see for your primary PCR that you have a positive control here and a positive sample which matches the band. Sometimes, however, you can see these really distinct banding patterns in your primary PCR, and what you want to do is run a nested PCR that looks at a narrower region of the mycoplasma. And over here, you can see that for these samples with the banding pattern, you get bands down here suggesting that your samples are positive, whereas over here, with these banding patterns, that sample is negative. So it's always kind of best to take another look with the nested PCR. With DAPI staining, I guess this image isn't entirely clear, but what you would see is a little ring around your cell nuclei, suggesting that there's micronuclei on the cell membrane. So a quick summary on mycotesting methods. PCR is incredibly sensitive, whereas DAPI staining has medium sensitivity. Staining may not be detectable at low-level contamination. PCR can be medium with specificity because you get these nonspecific binding patterns, but if you conduct a nested PCR, the specificity is pretty high. Unfortunately, mycoplasma is not the only kind of contamination you need to worry about. Bacteria can happen usually due to poor technique or contaminated media. Signs include cloudy media or a change in media color. You can see tiny blue dots under the microscope, and at higher magnification, you can see sort of these rod-shaped colonies forming. Fungus and yeast contamination can also occur. Fungal contamination is often sporadic, right? And it's usually due to a change in AC or vent system or changes in the season. You can see fuzzy, visible clumps in your flasks. And when this happens, it's usually best to toss your flasks and spray your incubators down with Spore Cleanse antifungal spray. Yeast can also occur and are visible as white circles in your flasks and can produce a mild odor. Another kind of contamination is contaminations with other cell lines. These are most commonly with 293s, and you can see formations of morphologically distinct colonies, changes in doubling time, and this is usually caused by working with multiple cell lines at once. Some ways to avoid contamination, of course, are to use sterile technique and to toss contaminated or suspicious reagents. Don't waste your time trying to figure out where the source of the contamination is. If you know that a certain media was used on a contaminated cell line, it's usually just best to get rid of it. It's also best to work with one cell line at a time in the hood, and it's best to thoroughly spray down your surfaces with 70% ethanol. It's also very important to dry your media bottles or reagent bottles from the water bath before you spray them down. It's also best to deep clean your biosafety cabinets with 10% bleach 2 times a year and autoclave your incubators twice a year or after a major contamination. I'm going to move on into sort of other additional best practices for cell culture. It's really important before you even start any kind of assay development to get to know your cells. After you thaw them specifically, it's best to take a picture, and that can help you understand what your initial morphology was and track any changes that might happen during your screen. For example, you'd want to look at signs of senescence, morphology changes, post-infection and drug dosing. You want to see if the cells change in size or if they're going from inherent to suspension. It's also best to check your incubator. Make sure that your CO2 and O2 levels are calibrated, best to do it monthly. Check to see if your shelves are even, because that could affect the way your cells are distributed in the flask. And you want to make sure that you're using appropriate volumes of media in your flasks. For T25s, it's best to use 5 to 10 mils. In T75 flasks, 15 to 25 mils. T175s, 30 to 50 mils. And in T225s, 40 to 60 mils. Now that I've sort of talked a little bit about contamination and tissue culture practices, I'm going to jump ahead into talking a little bit about reagents, specifically lentivirus. Lentivirus is best for transduction because they mediate potent transduction and stable expression into dividing cell lines and non-dividing cell lines in vitro and in vivo. They package their own reverse transcriptase and integrase, which can allow for seamless integration into the host genome and translation of their target proteins. To go a little bit into the virus production process, you want to have your envelope plasmid, your packaging plasmid, and your lentivector construct, that has your target gene. You want to transfect them into 293s and harvest your viral supernatant. GPP specifically uses the packaging plasmids PSPax2 and BSVG, and those are available through GPP. A couple notes on how to culture 293Ts for best viral production. It's best to split them 3 times a week, maintain proper density. They shouldn't be too sparse or too dense during your entire production timeline. And it's best to keep them at a passage number below 17. GPP's virus production pipeline is as follows. We aliquot our transfection reagents, which include OptiMEM, pDNA, and LT1. We transfect them into 293 T cells and change the media 6 to 8 hours after. We incubate that for 48 hours, and then we harvest and aliquot our virus. GPP can harvest productions up to 40 flasks. We have 5 members on our production team, and we have weekly production cycles. We produce genome-wide libraries and custom pools, and if you have any questions, you can contact Olivia Barre or Amy Goodale. So once you've produced your virus, you'd want to use viral titer as a QC metric. Titer attempts to answer the question of how many infectious particles you have per unit of volume, and with mammalian cells, titer is calculated as viral particles divided by mils. Determining your titer, however, can be very dependent on your experimental conditions. We've talked a little bit about your MOI, or your multiplicity of infection, and when it comes to titer, that's usually measured as viral particles per cells. But the number of active viral particles can be very dependent on your experimental conditions, thus so is your MOI. I'm going to go over a couple examples of why experimental conditions can affect titer. In this example, say you diluted 0.05 microliters of virus onto 1 million cells. You selected, waited for your uninfected cells to die out, and you stained the colonies that remained. Say you got 20 colonies per unit, you end up with 4 times 10 to the 5 infectious particles per mil. Say you ran that same experiment on a different cell line and got 400 colonies. Your titer's going up. Say that you added polybrine at the time of your infection and spun your cells for 30 minutes and got 10,000 colonies. Now your titer has gone up exponentially. So with these examples, I'm just trying to say that with the same virus, you're getting 3 different answers. Titer is relative to your experimental conditions. Now that you have your virus and you want to start planning your screen a little bit, Kendall mentioned that there are 2 screening approaches, the single vector, or your all-in-one library, and your split vector. Here are some considerations that you need to think about with each system. With the single vector, you're only doing 1 infection, 1 manipulation of your cells, and you're only using 1 selectable marker. However, because of the size of the construct itself, you're going to get a lower titer of your library. With the split vector, you'd be doing 2 infections, 2 different types of selection, but your sgRNA library can be made at a higher titer. However, long-term expression of Cas9 can be difficult. As Kendall mentioned, both of these vector approaches perform comparably to one another. So it's important to, again, get to know your cells and understand, based on their infectability, based on their growth, which system is more appropriate to test with. I'm going to go over just a brief schematic on how to make a Cas9 line. On the first day, you want to infect your cells with Cas9 at 1.5 million cells per 12 well. On day 2, you want to pool your cells and add blastocidin, and you want to seed a 6-well infection rate confirmation assay that can tell you that your non-infected cells have been dying at a particular dose of blastocidin. On day 5, you can check your confirmation assay to assess whether your selection is complete, but with BLAST, it's better to allow for 1 trypsinization before you count your assay. After selection, you want to assess Cas9 activity, and after activity assay, you want to make sure that you're fingerprinting again and banking Cas9 positive cells. Going into the Cas9 activity assay, you want to infect the cell line with your Cas9. For CRISPR knockout, you want to infect with PXPR011. We have 2 versions, version 1 or version 2, and select with Puro. And then with version 1, you want to measure GFP knockdown at day 9, 12, and 15. So over here, you have the parental cell line with no Cas9 infected with GFP, and you can see that 99.2% of the population is GFP positive. For the same line that's Cas9 positive, you can see a shift into the GFP negative space at 80.5% of the population. So we would deem this 80.5% Cas9 active. It's important to test activity of your Cas9 because, as you can see here, Cas9 activity can vary significantly across cell lines. It's important to see that with blood lines specifically, you see great variability. As I mentioned, we have 2 versions of the activity assay virus for CRISPR knockout. Version 2 just uses a destabilized GFP, and it reduces your readout time to 9 days post-infection instead of having to take multiple time points out. GPP assesses activity by establishing a 50% arbitrary cutoff. Anything 50% and above, we qualify as passing and able to go on to screening. It's important to just assess what your activity cutoff is because low activity in cell lines can result in poor or no results. So a quick summary of what I just went through. It's important to do some initial QC. Make sure that you're getting your cells from trusted sources, authenticating and microtesting them, avoiding contamination, considering which vector approach is best for your screen, and it's super important to measure Cas9 activity. With that, we're going to take a break.
Hi, I'm Sasha, and I'm going to talk about the importance of assay development in your functional genomics screens, specifically mostly the pooled screening. So there's 2 areas for optimization for your pooled screens. One, I'll sort of talk as screening conditions. This is your cell densities, doubling times, selection dosing, things like that. A lot of what Brianna talked about for array screening. The other is your assay conditions. So that's determining your drug dosing. There'll be your FACS gating, things like that. Screening conditions, as many people before me talked about, the success of your screen really depends on thorough and careful assay development and getting to know your model system. So you're really going to want to spend some time figuring out what confluency your cells like to be at. Many cells don't like to be super dense, and some don't like to be super sparse. The morphology and how it changes. Some cells will change morphology if they get really packed in tight or if they are really, really sparse again. You're going to figure out the size of your cells. So you can determine that with your cells per flask or cells per centimeter squared. But you're going to want to have an idea of that because that's going to influence your seeding densities, as well as any immediate conditions specific for your experiments as well. To assess the doubling rate for your cell line, you're going to want to seed cells in a 6-well plate at different densities. Day 3, you're going to want to change media, and after 96 hours post-seeding, you're going to want to count the cells to assess the doubling rate. And as a general rule, you're going to want to use the cell density that reaches 90 to 95% confluence after the 96 hours to assess your doubling rate and use as your seeding density in the following assays. Obviously, if you have a cell line that's a suspension line or that is not going to become confluent in any way, or cells that form sort of clumps and colonies that are never going to get confluent. You're going to want to have to just adjust this. So a lot of these experiments that I'm going to show you, you might have to optimize for your particular cell line. So this is kind of what the doubling assay might look like. So you can see that if seeding at 1 million cells, that after 96 hours you get a 95% confluence, and given that, the doubling time is around 44 hours. Okay, so you're going to want to use this seeding density and keep it in mind for following titrations, kill curves, and sensitivity assays. The next is determining polybrine sensitivity in puromycin, exactly like Brianna talked about. The polybrine is a polycation that can help infection efficiency, but it also comes with a viability hit for some cell lines. So you're going to want to take that into account. Day 1, you're going to seed cells into a 12-well plate with different amounts of polybrine. Day 2, you'll harvest those cells and seed them in different, plates with increasing amounts of puromycin or whatever selection antibiotic you choose to use. Day 6, you're going to harvest and count your cells. And as a general rule, you'll want to use the highest concentration of polybrine that results in less than 10% cell toxicity, and the lowest concentration of puromycin that causes complete cell death. So picking your doses of polybrine and puromycin. So you can see at 4 micrograms per mil, you get some viability hit, but not nearly as much as 8 micrograms per mil. If you're still worried about this viability hit, you might want to go back and choose about 2 micrograms per mil. And then for puromycin, you can see that you need to use at least 1 microgram per mil of puromycin to cause complete killing. More to this effect, we've looked more into the proper selection of infected cells and making sure that you're getting actually complete killing. And so what we have done is an experiment using our P. rosetta, which has both a puro and blast cassette in it. And then we've infected cells with this P. rosetta. On day 2, we add puromycin or blast. And then days 5, 8, and 12, we harvest and flow for GFP positive cells. And this allows us to look for the minimum dose that leads to 95% positive GFP cells. So as you can see from these graphs with A375 cells, using, for example, 1 microgram per mil of puromycin, you don't actually see 95% GFP positive cells until around day 7. And so if your assay you're planning to use an assay shorter than that, you would want to potentially use greater amounts of puromycin to see that actual killing. So you're going to have to do some optimization to make sure that you are completely selecting for your cell line. A small but important point is how you store your antibiotics and your drugs. So the conditions you store them can have an impact on their effectiveness. We have found that if you freeze thaw puromycin versus keeping it at 4 degrees, you actually lose effectiveness. And so that 1 micrograms per mil that normally would kill all of your cells now does not have quite as good of a selection of efficiency. So we always recommend that you make aliquots of your drugs and avoid freeze-thaw cycles. And we also recommend that those aliquots should be an amount that you can use at 4 degrees within 2 weeks. We've found that if you leave puromycin and blast over 2 weeks in 4 degrees, you also lose some effectiveness. So it's important to keep all of that in mind, even just for storing drugs and antibiotics. The next thing you're going to want to do is a virus titration. Similarly to what Brianna said again, the amounts that I'm showing here are just guidelines. Obviously, your virus might be at different titers. You're going to want to take that into account when picking your virus volumes. For Achilles, we actually seed 3 million cells per well into this 12-well on day 1. You might find that your cells don't like to be that dense, or that your cells are really hard to infect. You might want to choose a different density for that initial day. Day 2, you're going to harvest and seed cells into your 6th well with puromycin at these differing doses of virus. and you're going to do that at 24 hours post-infection. Again, you might find that your cells do not like to be this dense for 24 hours, and so you might have to harvest 6 hours after infection into your 6 well plates, and then 24 hours post-infection, you'll add your pyramycin. So those are just minor things that you might have to adjust. You might have to adjust these protocols when you do that. So I'm going to show and so then at day 5, we'll do your counts of all those wells to determine your infection rates. But I'm going to show a little video of the actual titration itself so you can see in real time how this works.
Viral titration, day 1, infection. Collect cells, making sure to keep the final cell suspension volume at 14 milliliters.
The only reason that we do that is that it prevents you from then having to spin down your cells later. So it's just a time thing.
Resuspend cells thoroughly and count. Combine your calculated cell suspension with media to a final volume of 14 milliliters. Add polybrine to your master mix.
Obviously, that'll be the polybrine dose that you determine in your polybrine sensitivity assay.
Label your 12-well plate with the appropriate virus volumes. Add your master mix to the labeled wells, making sure to resuspend your master mix thoroughly. Add the appropriate virus volumes to the labeled plate. Place your plate in the centrifuge bucket. Place the bucket into the centrifuge, making sure it's balanced. Spin for 2 hours at 2000 RPM. After the spin, add 2 milliliters of fresh media into each well and incubate overnight.
So we do that to dilute out the polybrine. You may find that it's better for you to change the media completely to help prevent toxicity to your cells on the polybrine.
Day 2, 6-well seeding. Trypsinize your cells and count each well. Set up your 6-well plates for seeding. In one plate, add fresh media. In a conical, add pyramycin to fresh media, resuspend, and add the pyramycin media to your second plate. From your counts, add the appropriate volume of cells from each well into corresponding puro and no puro wells.
And that's going to be at the seeding density you determine in your doubling time assay.
Seed at a density that will allow the cells to select and proliferate in 72 hours. Day 5, readout. Resuspend and count all of your wells. Record your data and determine the virus volume and the number of plates for large-scale infection.
All right, so in order to determine the amount of virus that you should use for large-scale infection, you're going to want to consider MOI. So we want to have only one perturbation per cell, so like one guide per cell, and that's going to prevent noise in your infection, unlike with the arrayed screening, where you're looking for a high MOI. The way we've determined the percent of infected cells that you should use, which is around 30 to 50%, that's the infection rate that you should be aiming for, is through the Poisson distribution. And you can see that at between 30 and 50%, over 75% of your cells have one shRNA. Most of your cells are uninfected, but 75% of the infected cells have only one shRNA, and very few have more than one shRNA. So that's what we're kind of aiming for, and that's why we look for an infection rate of 30 to 50%. So this is an example of what your day 5 results might show. So you can see that with the minus puro cell counts, you're going to determine your viability. So that'll determine if your virus is causing any sort of cell toxicity, which can sometimes happen, especially at these higher doses. The plus puro cell counts are going to give you your infection rate, and you can see that at between 75 and 150 microliters, we're getting that 30 to 50% that we're aiming for. So that's where we would aim to, that's the amount that we would aim to use for this particular cell line with this particular virus. Representation. We've had multiple people today talk about representation. So as Federica has talked about, we aim for 500 cells per perturbation, and so, for example, the Ivana library has 80,000 guides times the cells per guide, which is 500. You get 40 million cells that you're aiming to have infected. With positive selection, we like to have more than that. We like to have a higher number of cells per guide, and so if you do that, you'll need more infected cells, and you're going to want to use the results from the virus titration with the infection rate and your viability to determine how many cells you need for your infection, along with the number of replicates you're choosing to do to maintain representation. So I want to give a little example of how important it is to really be doing all of these experiments and keeping in mind these numbers when you're doing your assay dev. So, for example, if you seed your cells on day 2 in the 6-well plate for this virus titration with 1 million cells per well, and this particular cell line has a doubling time of 44 hours. On day 5, you do your counts, and you see it looks like really nice. You get this like 30 to 50% range. Like it looks like you're good to go. But if you look closer, at this range, if you look at the minus pure well, with a 44-hour doubling time and having seeded 1 million cells, you should have actually expected to get 3 million cells here instead of 2. And so if you calculate out the doubling time there, the doubling time is actually 80 hours. And that would actually indicate that you misseeded up here, that this was not the right amount to seed in this well for this assay, and that your cells are now completely overconfluent, and that this is now artificially inflating this infection rate. So if you went and took this amount of virus and infected your cells at large scale, what you would find is that you'd actually get an infection rate of 10 to 15%, and that would probably indicate that you would not have enough representation. You might have to scrap your screen, as opposed to if you had been keeping all this information in mind when you got this result, you would have gone back and redone your titration with the proper seeding density and would have prevented this from happening, and you would have saved a lot of time and money. So the next thing I'll talk about are optimizing assay conditions. I'm going to focus mostly on like drug sensitivity assays as opposed to any sort of in vivo assay development that you might want to do. So for that sort of stuff, I would definitely talk to a screening scientist, but this is just an example of the amount of work that goes into assay development for your assay readout. So this requires a long period of time generally to optimize your assay conditions. You want to take a lot of time up front to fine-tune your drug doses, your flow cytometry grading, the length of your viability assay, et cetera. We even suggest that if you can repeat your assay optimization either in your hands or more ideally in somebody else's hands, that can further enhance your chances of success if you're able to see that happen. So this is an example of how you would do a drug sensitivity assay. Day 1, you infect your target cells with a control lentivirus, such as GPP, and try and achieve a 30% infection rate. You might have to do a titration with this virus beforehand before doing this drug sensitivity assay. On day 2, you're going to harvest and plate your cells into T75 flasks in the presence of Puro. You're going to want to take that seeding density and extrapolate the cells per centimeter squared to seed into these flasks. Day 6 after Puro selection, you're going to split the cells into T75s in the absence of Puro to let them recover from selection. 24 hours later, you'll add drug. You'll have a DMSO arm that doesn't have any drug, and then you'll have increasing amounts of drug, your drug of choice. Obviously, this might not be the appropriate amounts for your drug, so you'll have to determine that yourself. From day 10 on, you're going to want to count and passage your cells every 3 to 4 days, or depending on your doubling time, that might change, but generally every 3 to 4 days for 2 weeks to determine the cumulative population doublings in each condition. This is an example of what you might get. So you can see that at the earlier time points and at the lower drug dosings, you don't really get any window between your DMSO arm and any of the drug control arms. So you're looking for a good signal-to-noise ratio between your drug arm and your DMSO arm. However, these assays don't always turn out quite so nicely. I'm just going to give a couple examples of some of the further optimization you might need to do after doing this assay. So for this assay, you can see you have a low potency drug. The initial optimization done, the initial curve done, resulted in a really small signal-to-noise ratio. And if you went forward with the screen, you have a high risk of not being able to see any hits because of all the noise. However, we optimized this assay by maintaining the cells at a low density and refreshing the drug every 3 days. And we ended up increasing this signal window quite substantially, and it resulting in a really successful screen. You also might have a really high potency drug. So these, before optimization, you just saw complete cell death, not really able to get any cells out at the end, so not really able to do the screen at all. What we ended up doing is, after initially increasing the seeding density, we reduced it a little bit and didn't passage the cells quite as often, and instead did some media changes. And we were able to prevent this cell death and result in a successful screen once again. And if you're really spend the time up front to do all these optimizations, you can even see that using this cell line with this same drug and holding these screening conditions, that you can go across libraries and still get successful assays, which is pretty awesome. Some final thoughts. As this old house says, measure twice and cut once. Take the time up front to do all of these optimizations, and that will ensure maximum success in your screen. Save time and money, a lot of time and money in redoing your screens if you don't do this. Optimization is key. Like I said, there's all these little things that you're going to have to do for your particular model, your particular cell line. These were all of these assays I showed you were just general guidelines. You're going to have to do optimizations based on how sensitive your cell line is to density and things like that. And then use your resources. The GPP screening scientists are really good at figuring out what you might need to do next for your assay development in terms of shifting your seeding densities, and so utilize them as much as you can. So now that you've optimized both your screening conditions and your assay, you're ready to begin some screening, and I'll hand it over to Yanari to talk to you about that.
So hi, I'm Yanari. I'm going to be talking to you about how to navigate viability and positive selection screens. So a general overview, what we're going to be seeing is that we're going to be taking your cells and infecting them with our pooled library, and then what we're looking to get is a low MOI transduction. From there, we're going to select our cells and apply selective pressure for it, and then we're going to divide into either positive selection screening passaging or to viability screening. And then we'll do passaging that way, and then once we're done, we'll then send the cells for samples for gDNA isolation. Our general timeline for preparing for large-scale infection is going to be day 0, which is going to be our preparation for a large-scale infection. This can take more than just one day. Then we'll proceed to large-scale infection. Day 2, we will pool and seed our infection rate confirmation assay and also split into replicates. Then on day 5, we'll read out our assay, and then from there, we'll proceed to passaging in either a positive selection or a viability screening mode. So to prepare your cells for large-scale infection, you need to first look at how many cells you actually need for this. So as mentioned before by Federica and by Sasha, we're going to take our cells per perturbation, times that by the number of perturbations we have, and this will give you our total cells to start with. We then want to take this number and divide that by our expected infection rate and our expected viability, which will give us our actual number of cells we need minimum for infection. So for example here, we're going to take our 500 cells per perturbation, as we said before, or 80,000 perturbations, and we get 40 million cells. Now from our previous assay dev, we know that we should be expecting 40% infection rate and maybe no viability at all. Then we need a minimum of 100 million cells to start with. Were they going to add a 10% overage to this? And the reason for adding overage is that you have to remember we're scaling up from assay dev, which was just a one-well infection, to many, many plates now. So we want to add 10% overage because we're going to expect to get a little bit of loss in infection rate and loss in cell viability. That gives us our actual number of cells we need for this infection. We also want to make sure, we want to know how many flasks to expand up to when we're doing this infection, so we're going to divide that by our cells per T175 to give us how many flasks we need for expansion. So if we needed 100 million cells and we had 10% overage, we get 110 million cells. We know we can fit 50 million cells per T175. That gives us 2.2 T175s to expand up to. Since we can't see 0.2 T175s, we're then going to just round that up to 3 T175s. So 110 million cells into 3 T175s. This, again, gives a little bit more insurance as well to make sure we have more than enough cells for infection. Now that we have our cells ready for infection, we can proceed to large-scale infection. So we're going to make a first master mix that contains our media, cells, polybrine, and virus. We're going to mix it well, plate out our master mix, and then put it in the centrifuge for spinning. When you're creating your master mix, it's really important you actually follow this guideline here, where you add the media first and then the cells. Before you add your polybrine, you're going to want to make sure you actually let your virus thaw out first. And when your virus is mostly thawed, then you want to add your polybrine. The reason for this is, if you're letting your cells and media sit in the polybrine for extended periods of time, this can actually attribute to your cell viability when you're counting out your reps and proceeding to your passaging. So to prevent that from happening, we're going to let our virus thaw, add our polybrine, mix it well, and then we're going to seed our one well of no infection control well. Do not forget that. That's incredibly important. Once you've added your polybrine and seed your no infection well, then we can add our virus and then seed out the rest of our cells here. I also have a short video to actually show you how we proceed with large-scale infection.
Large-scale infection, day 1. Collect cells from your flasks into a conical. Resuspend your cells thoroughly and count. Make a master mix by weighing out media and pipetting your cell suspension. Make sure to resuspend your cells thoroughly. Add polybrine to your master mix and gently swirl the bottle to ensure that everything is thoroughly mixed. Label a no infection control or an NIC well. Add 2 milliliters of the master mix to your NIC well. Add virus to your master mix at the volume determined by your viral titration. Mix the final solution thoroughly and plate 2 milliliters of the solution into each well of your 12-well plate. Spin plates for 2 hours at 2,000 RPM. After the spin, add 2 mils of fresh media and incubate overnight.
So you can see in the video there that we actually were measuring out our media with the scale. We generally do that because it's easier than pipetting out, 260 mils of media. 1 gram is usually 1 mil, so we just stick with that. And as Sasha mentioned beforehand, if your cells are particularly sensitive to polybrine, to virus, if you're worried about them, you might end up doing an entire media change or harvesting the cells earlier. So we're going to proceed then to pooling. So in this example here, let's say we have 8 plates total. What we're going to want to do is we're going to put into different replicates. So for this example, we're going to say we have 2 replicates, rep A and rep B. So we're going to take 4 of our plates over 8, pool them together, count them, seed for infection rate confirmation assay, and then seed the remaining cells as a replicate A. We're then going to take the other 4, pool and count them, seed our infection rate confirmation assay, and seed the remaining as replicate B. We're going to take our 1 mil of no infection control cells, count them, and seed our infection rate confirmation assay. I have another short video to show how we proceed with pooling.
Pooling large-scale infection. Collect the cells from the NIC into a conical. Collect the cells from the wells and pool into replicates in separate conicals.
You can see we have our separate conicals here, rep A and rep B already.
Spin the conicals at 1500 RPM for 5 minutes, aspirate off the media, and resuspend in fresh media. Resuspend the cells thoroughly. Count the pooled cells for each replicate as well as the NIC well. Plate an infection rate confirmation assay with puro and no puromycin wells. Seed the cells into the plate at the density specified by the viral titration. Include the NIC and the replicates. Reseed the remainder of the cells from the replicates into the appropriate number of flasks. Add fresh media to each flask, bringing them to their final volumes. Add blastocidin to each flask. Add puromycin to each flask. Mix the flasks gently and incubate.
So the important thing to notice is that we already split into replicates, and from that point onwards, you're going to want to treat each replicate as like a different cell line entirely. Don't let the two intermix. Don't try to mix them together and then split them later again. Now we talked a lot about this layout, the infection rate confirmation assay, and this is the layout that we use. So we have our no pure wells here and our plus pure wells here. Our no infection control cells are replicate A and replicate B. Now you'll be seeding in the density that was previously determined in your assay dev, and you'll be using the pure concentration that was also determined in your assay dev. If you do not know these numbers, you should not be proceeding to this step at all. You should be still in your assay dev phase. It's really important. So the other thing you can do is you can also calculate on day 2 what your expected representation is. This is going to give you an idea of what to expect for your day 5 readout and lets you know if you're going to be okay to proceed with your passaging as well. So to calculate for that, you're going to take your cells recovered, take your times that by your expected viability and your expected infection rate, divide that by your library size, and that will give you your cells per guide. So if we're expecting a 90% viability and a 50% infection rate, we should be getting 562 cells per guide. And remember, our target cells per guide was 500, so that puts us in a very good place. If you're good for your representation, you then want to proceed into knowing how many you're going to seed into, how many flasks you're going to seed into. This is also really important. Seeding too densely might mean that your selection drugs might not have a chance to actually work, and you might actually get an inflated infection rate when you do your readout. Likewise, if you seed too sparsely, you're going to not really give your cells a chance. You're putting them under a lot of stress with the puro and also the transduction. You can actually lose a lot of your cells or get a really low infection rate because of that. So to calculate for your number of flasks to seed into, you're going to take your cells recovered, times that by your expected population doubling, times that by your expected infection rate and your expected viability. Divide that by your cells per T175. So we're expecting 50% infection rate and 95% viability, and we know that we can fit 50 million cells. We're going to get 2.6 T175s. And again, you can't seed 0.6 T175s, so we're going to take all our cells and seed them equally into 3 T175s instead. If you're finding that you need to seed into a lot of flasks, like 7 or 10 T175s, there are other plastic wares out there available. The most common ones that we use in GPP are 2 layers and 5 layers. A 2 layer is about 7.5 T175s, and a 5 layer is 18 T175s. And there's some considerations to take when you're using these layers. So the benefit of them are that they're obviously easier to passage than passaging 10 or 12 T175s, and you can fit a lot more cells into them. That said, you will be expecting some percentage of cell loss, and a lot of this is due to the fact that the more layers you have in these plastic wares, there's actually less oxygen CO2 flow through them. So you might get some cell loss there, and they're just generally hard to get all the cells out of since you have multiple layers. Another thing to consider is that if you're using something beyond a 2 layer, so anything above a 2 layer, they actually don't fit under a microscope, so you can't see them. So you're going to want to seed a side T175 at the same ratio as your 5 layer to see what your cells look like. We generally recommend using these layers only for viability screening and for adherent cell lines. We don't recommend these for positive selection screens or semi-adherent and suspension cell lines. Now that we've seeded our infection rate confirmation assays and our replicates, on day 5, we're going to do the readout of this infection rate confirmation assay. And this gives you a lot of information. This is almost like a miniature assay dev readout for you as well here. So what you can see is on the top row over here with our minus pure, you're able to see a viability effect by comparing the NIC to the REF A and REF B and the non-pure wells. You'll also be getting your infection efficiency between your replicate A and replicate B when you convert them to minus pure wells. And lastly, you'll be able to see your pure efficiency through this. When you're doing this readout of the plate, there are some important things to take into account. So you want to make sure your no infection control wells are fully dead at this point. And you can't really assume that by just looking at them. The only way to really make sure that you have them all fully dead is to do a trypan count. And especially if you're working with semi-adherent suspension cell lines, you have to be doing trypan counts. They might look dead on the scope, but there's no real way to figure that out unless you actually do these trypan counts. So please make sure you do them. The other thing you want to do is you want to do more than just a comparison by eye. You want to count each well. Of course, you'll look at the wells under the microscope, and you'll see that they're about 50% or so, but you don't really know for that for a fact until you actually count these wells physically and compare the numbers together. You then want to compare your answers to your assay dev results, and they should pair up pretty well within 5 to 10% of what you got for assay dev. Keep in mind, though, that if you have a higher infection rate, this does not actually mean that you have a better screen. Likewise, if you have a lower infection rate, it doesn't mean that your screen has failed either. And once you've got your infection rate, you want to make sure you recheck your representation at this point, since you now have your actual infection rate and your viability effect. So to do that, you're going to take your cells recovered from your replicates, times that by your infection rate, and times that by your viability effect. Divide that by your library size, and that will give you your cells per guide. So keep in mind, our targeted cells per guide here is 500. So in these two examples here, we have one that was 45% infection rate and 95% viability. We have 481 cells per guide, which is very close to our 500, which is within 5 to 10%, so that's good to go in the two replicates. Now, let's say our infection didn't turn out so well. We have 30% infection rate and 75% viability hit. We're left with 253 cells per guide. That's about half of what we were actually aiming to get. Even though you might be tempted to toss your screen and redo it entirely at this point, there are other options. So if in our example we have two replicates, we can actually collapse rep A and rep B together into one replicate, and this boosts our cells per guide a little bit higher and allows us to continue with one replicate. I do believe Scott will go into this a little bit more in detail for post-screening processing later. Now we're going to move on to actually passaging our screen. So we're going to switch to viability screening first. We'll touch on positive screening after this. So when you're doing viability screening, this is actually quite a regimented schedule, sort of passaging schedule to be seeing. So we're going to be expecting to passage our cells every 3 to 4 days, or doing a media change every 3 to 4 days. You should be getting enough cells at each passage to pellet them down and still seed for your passage. And on top of that, you should be expecting a normal, very regulated growth curve. There shouldn't be a lot of variance at this point. So when you're passaging your cells, what you're going to do is you're going to take them, so rep A and rep B separately. You're going to harvest and count the cells, and then you're going to seed back at representation into your flasks, and then you're going to pellet the remaining cells. Do not toss those cells. The reason we don't want to toss these cells is because when we have all these pellets from every single passage, it gives us a really great timeline. So if you're finding that at the end of your screen, you're like, oh, I want to go back to earlier passages, you have these on deck to use them later on. You want to make sure you're recording all your data, and I can't emphasize that enough. By recording all your data, I mean you want to be writing down somewhere how many cells you got out for passage, how many cells you're seeding back, how many cells you pelleted, anything that you might know that's a little bit funny or off. These are all a way to keep track of what's going on with your passage and if something is going wrong or not. So in this example here, this is an example of a screen we did in Project Achilles. So here we have days in screening, and here with the cumulative population doubling. So the red line here indicates what we were actually expecting for a growth curve for the cell line. This is a cell line that doubled every 4 to 5 days, so we were expecting a slow sort of growth. When we actually did this in screen, in the blue line here, you can see that the actual doubling rate increased. And by the end of the screen, we were 2 cumulative populations apart. And we were kind of like, that's not what we were expecting. When we sent these end samples for fingerprinting, what we thought were CAPN2 cells actually turned out to be HEC293T cells. And the reason we were able to have an inkling that something was off was because we recorded these doubling hours and we knew what was going to happen with these cells, and we were getting something different from that. There are some other warning signs as well when you're screening to look forward to, or not look forward to keep in mind. Don't look forward to those. So as I mentioned before, doubling time is an indication that you might have cross-contamination of different cell lines, and Zora had previously shown a great image of what these two might look like. If you notice an increased debris in your media and a lot of cell death all of a sudden, this can be a sign of contamination, or on the other hand, it can actually be a sign that you seeded your cells too sparsely and you need to condense your flask at this time. If you notice a change in media color, again, this could be a sign of contamination, particularly mycoplasma, or this could be a cell line that you actually just need to media change more often. Instead of maybe every 3 to 4 days, maybe it's every 2 days, maybe it's every day. And lastly, if you're noticing morphological changes, this can be a sign of cross-contamination of different cell lines together. So again, you want to be making sure you know what your cell lines look like, taking an image beforehand, having all these in hand to make sure you know what to do with them. And again, we can't emphasize enough that you want to make sure you're myco-testing as often as you can, and that you do send your samples for fingerprinting at some point to see if you have the cell line that you actually think you're screening with. When you're at the end of your viability screen, you're going to take all of your cells, rep A and rep B separately, spin them down, aspirate the media, resuspend the PBS, and then you're going to divide them into multiple vials. You want to divide them into the vials at representation. So as a reminder, representation is cells per perturbation times that of your perturbation, and that will give you the number of cells you want to pellet at. So if we have 500 perturbations, 80,000 perturbations in the library, you're going to want to seed at 40 million. So let's say we had 120 million cells at the end of the screen. You'll have 3 vials you want to freeze down in. The reason you want to do multiple vials at this point is that this ensures you some flexibility. If something happened post-screening process and you lost a vial, you have 2 more to go back onto. Or if you needed more vials for gDNA, you have all these vials to go back onto. Do keep in mind that certain cell lines will require longer readout time points. So for Project Achilles with their Ravana library, we typically screen for 21 days. However, if you're screening with an epigenetic library, you're going to pull that out to about 4 to 5 weeks to allow for all the changes you want to happen appropriately. We're now going to move on to positive selection, small molecule passaging. And the first thing I really want to point out is that this is quite a shift from viability screening. There's no real set template. And this kind of really highlights the fact that you need to have a very good assay dev to proceed with this passaging. Your assay dev is what's going to tell you how to passage, how much, how often, what density, what you're going to be expecting to see. If you don't have an assay dev, there's no real way to proceed with positive selection. This is not something you can just kind of decide on a whim. So this here is an example of a screen we did for the drug resistance project in GPP. And what you can see here are these really nice little colony formations here, and this is a sign that these cells are beginning to resist the drugs that we put onto them, and the other cells are dying out around them. This is generally a sign that something good is happening in the screen. That said, don't rely just on this. We have had many screens that are successful without these colony formations as well. But this is generally a sign for us that we know something is happening and something is good is happening. This is an example of what sort of data we're going to be seeing during our screen by all the recording we're going to do. So here we can see our DMSO arm growing as we expected. We have our DMSO negative control arm here in purple. And the negative control arm is really just an arm that we infect with virus, but no perturbations in there. So it's just showing us what's happening. Here in the orange, we have our drug negative control arm, and we can see that it's starting to die off at this point over here. But our drug arm over here in the green is showing resistance and outgrowth. And so because we recorded all these data points here and we can see this starting to happen, we decide that this is a good point to end the screen. And this is kind of what we want to see. And around this point is when we can begin to see those colony formations. But again, you might not see that if you're doing your screen. All of these little nodes here denote passages. And again, we decided when to passage these things by our assay dev. So our assay dev is telling us how to do these passaging, when to do it, and what point. As much as I say you need to follow your assay dev, there may be times when your cells don't exactly follow your assay dev, and there might be some differences. And remember that when you have differences between your assay dev and your screen, it doesn't mean something's going wrong. You have to remember that you're scaling up from really small size to a large scale. And most importantly, you're introducing perturbations at this point, which you didn't beforehand when you're doing your assay dev. So there will be some changes between your assay dev and your large scale infection. Some things we have noticed beforehand, you might see a change in morphology. And when I say change in morphology, this is not the same thing as you would expect for contamination. This is when you put cells on drugs, you might see that cells become very, very large and expanded. They might become very, very skinny. This happens with drugs. The best way to go about to how to deal with that is to then readjust your cells per centimeter squared, and you can either increase or decrease your flask size accordingly. Hopefully this is something you saw in your assay dev since your cells would have been on drug as well at that point. So you should have an idea of how many cells per centimeter squared to work with. Likewise, if your cells are not dying as you expected, You may need to readjust your cells per centimeter squared and increase your flash size. You also want to consider passaging more often, because sometimes passaging more often might kickstart these drugs a little bit more for you. You also want to be double checking all of your stocks. So you want to make sure that all of the drug stocks you made, all of the medias you made, everything you're using is at the actual right dosage you want it to be. You don't want it to be a technical error that's causing these things to happen. So make sure you do that. Likewise, if your cells are dying more than you expected, you are going to want to passage all the cells or only media change, and you want to avoid pelleting any cells when you're at this passaging phase as well. You can also potentially consider ending the screen earlier. And again, you want to double check all the stocks you're working with as well. When you are at the end of your screen for positive selection, again, you're going to take all your cells, spin them down, aspirate them, receive them to PBS, and you're going to pellet your cells. Now, the difference between here and viability screening is that you actually might not have your cells at final representation, and you might have a lot less cells. Don't panic if that's the case. Remember, this is a positive selection screen, so you're expecting certain perturbations to outcompete other ones, so you might have less cells than you actually started off with, or less than your representation is at. Just make sure you pellet all of them into one vial, and you are good to go for your post-screen processing. So an overview of what we just did, we prepared ourselves for a large-scale infection, proceeded to our large-scale infection. We pooled and seeded our infection rate confirmation assay, as well as splitting into replicates. We then read out our assay and proceed to our passaging in either positive or viability screening. And I'm now going to pass it off to Scott to talk about post-screen processing.
Thank you. Okay, so I'm Scott Younger. I'm a screening scientist here in the platform. I'm going to talk about kind of the end of your screen and wrapping things up. Do you have a question? Yeah.
Or a optimized media, same media for every experiment?
So for viability or positive selection?
For all of these.
So your media conditions should have been predetermined in your assay depth portion where you optimize on your media for your cell lines.
So it is specific. Each cell line gets its own.
Yes.
Okay, so I'll be talking about finishing your screen. I thought this picture was appropriate because you've gone through this idea of planning, or had a concept for a screen, then you plan the screen, and as you know now, you've done a lot of work in executing the screen, and now you're to the points where you get some results.
Should we take anything out of the fact that you're like still way down at the base of the top?
I actually did not go up there. So this is Maroon Bells. This is southwest of Aspen, Colorado. It's actually Twin Peaks. So I figured this is your results, and then this second peak is your validation, which is something for another day. Okay, so you've gone through the process of taking your virus, infecting cells. You've done the screen, whether positive or negative selection. And now we're going to focus on this part, which is isolating the genomic DNA from the cells you've screened. We're going to amplify out barcodes. So I'll use the term barcode or element or guide RNA. Those are all synonymous when I'm talking about this topic. So we're going to amplify those out of the genomic DNA you've harvested. We're going to perform next-gen sequencing, and then we're going to align those reads, or extract the barcodes, align the reads, and see how your screen went. Okay, so first to focus on isolating genomic DNA. So to do this, we use these column-based genomic DNA purification kits. There's several of these commercially available. We use the NucleoSpin Blood XL kit. There's ones from QIAGEN and a lot of other sources. So they all work basically the same way, where you take your cells and then you lyse them. You mix them with some proteinase and RNase to get everything out of there, all the other gunk. You bind the DNA to a column, wash it, elute it off. So it's pretty straightforward. And when you're doing this, you should just follow the protocol from the kit. One thing we want to emphasize is that you need to practice using these kits before you're ready to do your screen. Because you don't want to be at the endpoint and find out that you need to use buffer AE1 and buffer AE1.1, and you don't know what the differences are. And you don't want to be figuring all this out at the same time you're harvesting these cells that you've just been screening for months on end, potentially. Okay. So there's a video that we'll add later that you can see that protocol, but really you should go through the protocol for the kit that you're using. So next we'll focus on amplifying barcodes. So the way this works is here is your viral vector. This is what we use to actually make the virus. And if we zoom in on the sequence here, what you'll see, the U6 promoter, this is the guide RNA, and this the tracer RNA. So for the different technologies, this may be a shRNA or it may be an ORF. It may vary. So what we do is we target these regions with primers, and appended to these primers are the Illumina adapter sequences, the P5, and we use an indexed P7. So when we perform the PCR, you're going to get a PCR product for the guide RNA vectors. That's about 350 base pairs. That's going to vary depending on the screening technology you use, whether it's an ORF or an shRNA. And this PCR product is what we're actually going to sequence to see what guides made it through your screen. Okay. So one thing we really want to focus on, yes? So we typically do 28 cycles of PCR. That's kind of our standard. For certain screens, like some positive selection screens where you have very, very little material, we can extend that to longer. That's or more cycles. That's something you should talk to us directly about when we're kind of going through the details of your screen. Is you have to be concerned about over-amplification and jackpotting in that situation, but you also need something to sequence. But 28 is typically what we do. So an important factor is the amount of template gDNA. So everybody in this room, or a lot of you have probably done PCR, and I'm sure a lot of you have done PCR from tubes where there's like literally nothing in it or trace amounts of DNA. And that's not what you're going to be doing here. You actually have to calculate the amount of template genomic DNA you're going to use. And the way we do that is we take the number of elements in the library, we multiply that by the representation you were anticipating with number of cells per guide, and then we multiply that by the gDNA content of each cell, which for a million cells on average about 6.6 picograms of genomic DNA. And it's going to tell you the amount of genomic DNA required for PCR. So an example calculation, so our Avana library, which is 80,000 guides, if we were going for the 500 cells per guide representation, it's 6.6 picograms per cell. You're going to need to perform PCR from 264 micrograms of genomic DNA. So it's a lot of genomic DNA, and it's more than you're probably used to handling if you're not doing these types of screens. So when we do these, the kit we use in GPP is this Xtera or Takara XTAC kit, sorry. And the maximum gDNA we can fit into a reaction for this is 10 micrograms of gDNA. More than that, it's going to kill the reaction. So if you do the math of needing 264 micrograms of DNA and you can only fit 10 per reaction, we round that up to about 27 reactions that you're going to need to do from your template genomic DNA. So it's not a single reaction. We have like 27 for a standard screen of this size. Okay. So we do PCR and 96 well plates. And so the rest of what I'm going to go through is kind of how we handle plates in the platform. And if you screen through us, you'll be submitting plates to us, and this is what will be going on when we're handling your plates. Okay. So the plates are 96 well skirted plates, a maximum of 10 micrograms of gDNA per well, and we have a fixed volume when you submit a plate to us. It needs to be in 50 microliters. That's a fixed amount. Okay. So from the previous example where you needed to have 27 reactions, you would just fill the first 27 wells with the gDNA from a single sample. Now if you have more than one sample, you just add an addition, say to the same screen, say another replicate, you would have 27 more wells. So for 3 samples, 27 more wells, and say you had a fourth. That's clearly not going to fit on this. And this would be a situation where you're going to submit 2 PCR plates. And if you're going to do that, we ask you to reference the submission guidelines, which you can get by emailing us, or it's also available on the website, because there's some considerations for how you're going to want to do a plate layout if you're going to be submitting multiple plates. Okay. So this is what happens when we see your PCR plate. You'll notice that all the colored are gone because we don't know what's in your plate. We just know that there's samples in all of those wells. And we know that they should be 50 microliters per well. So what we're going to do is we're going to add a PCR master mix, which has all the things we need, like water, DNTP, PCR buffer, and the P5 primer. And we're going to add that at 40 microliters per well. This is why volume matters very much. And then we also have a P7 primer plate. And we take this directly out of a 96 well plate that has an indexed primer in every well. So when we perform the PCR, every well is going to end up with a unique P7 index. Okay. And then we add this, we take this plate, this is the plate that you submitted, we actually do the PCR right in that plate. Okay. And you'll see, based on these volumes, you need to submit in 50, otherwise it's going to mess up the PCR reaction. Okay. So post-PCR, we have a 96 well plate, and we have a barcode, a unique barcode on the P7 index, or P7 index for each well in the plate. So what we're going to do now is take all these reactions and we're going to pull them together into one tube. Okay? So from that tube, we're going to do cleanup purification, and we use SPRi beads to do this. If you're not familiar with these beads, you basically just take your DNA sample, you mix them with the SPRi beads, which will bind up the DNA. These are magnetic beads, so you can toss them on a magnet. You do some washing, and ethanol washes, and then you can elute the DNA off in a concentrated water or TE. So we're going to use these. We're going to take your pool of PCR products. We're going to go through this process, and we'll end up with pure DNA that we can use for next-gen sequencing. Any questions? Okay. Okay, so now on to sequencing. We sequence everything on a HiSeq 2500. This is, in the setup we use is 8 lanes per flow cell, and we get an average of about 170 to 200 reads per lane. The way we do this is we take one plate, which we pull together, and each individual plate goes on one lane of the HiSeq. Okay? And the way we have this set up is that we can split this over multiple screeners. You don't need to have 8 lanes to submit to a HiSeq. Say you have 2 plates, you'll go on a flow cell with other screeners that may have 1 or 3 or 1 plate. So we mix those all together until we have 8 plates ready for submission, which happens pretty quickly, and then we submit that directly through the genomics platform. And one of the nice things about the way we have this set up is that you get to enjoy the lower cost of sequencing, which is about $1,100 per lane, versus if you were to go walk up to GP and try and get a similar technology for sequencing, you're going to pay almost twice that. So it's really cost effective to have us handle your samples, process, and go through sequencing. Okay. Okay, so now the next part, aligning and counting reads. So we've gone through the process of performing PCR and pooling your samples, and then we sequence them. And then what comes out of that is a sequencing read. And there's some information here that tells us where this read came from. We know that it came from well A1 based on the barcode here. We have the actual sequence of the read and then some quality metrics. So if we focus in on the sequence of the read, you don't need to worry about the specific sequence, but this aligns to the U6 promoter that was in our viral vector. This is the guide RNA, and this is the tracer RNA. So what we can actually do for each read is extract the barcode, which is the actual sequence of the guide RNA. And then we know that that read came from, well, A1. So we end up with a table that looks something like this, that for each read, so up to 170 or 200 million reads, we know which well it came from, and we know what the barcode was. So we can count those up and aggregate all that. But the problem is that we know what well that came from, but that doesn't mean very much to us. We just know that there was 96 wells and a read matched to that well. So we need to know what those wells actually had in them. So when you submit a sample to us, you supply a conditions file. And this is something you can get from the website, or you can email us, and then we'll send you all the details. And what this sheet has is just some spots where you're going to put in the amount of DNA that was in each well, along with some meta info, which tells us samples and which wells had which samples in them. And that's important for aggregating reads. Specifically, so we have here's your sample, your layout that we know of now based on your conditions file. So what we're going to do is take all the reads that match the same sample, and we're just going to aggregate those reads. So that's how we handle that. Without the conditions file, we can't do that. Okay. So here's what we end up with. We have raw reads for each sample for each barcode. So here's just an example of barcodes or guide RNAs in your library. And here's how many reads match to that barcode in each of these hypothetical samples. So when you look at these numbers, you see that you get about the same amount of reads for this barcode, pretty different reads for the second barcode, right? So based on these numbers, you would think this was equal. The first one and the second one, you would think that was pretty high. But something you have to keep in mind is that you get a different number of reads for each sample, total reads. So if you count up the total amount of reads that match to each sample, you'll see that's not always equal. We actually got twice as many reads in this sample as we did for this sample. So what we have to do is normalize the reads. And the way we do that is by taking the total reads, the raw reads per barcode and divide that by the total reads, multiply that by 1 million. So this is basically reads per million, and we take the log of that. We also add 1 at the end of that because this number can actually be 0, and you can't take the log of 0. So this is what comes out in our normalized reads. Any questions on that? Okay, so here's a table showing what the normalized reads actually come out to be. So for this first barcode, you see there's actually quite a lot of variation in what abundance actually is in the library, versus the second barcode, which based on the raw reads look quite different. Turns out it was equally abundant in all the libraries. So it's important to normalize the data. When you screen through us, we'll send you files that contain all of the raw barcodes or raw read counts, and we'll also send you a file that contains the log normalized data too. Okay, so did it work? Now you've gone through this work and you have this table that gives you some log normalized read counts. So what we're going to do is go through and look at some key metrics to assess whether or not your screen was successful. And these metrics vary based on whether you're performing a negative selection or a viability screen, or a positive selection like a drug resistance screen. So for negative selection screens, we look at overall library representation throughout the course of the screen. We look at replicate reproducibility, both in terms of normalized read counts and log fold enrichments. And then also another important metric is depletion relative to representation. I'll go through what these mean. So library representation. So all of the elements in your library for a negative selection screen should be detected throughout the screen. I say all, but the vast majority, we can say maybe. So here's just an example of a screen where this is an early time point, and this is sample 1 or sample 2 at the end of a screen. And you'll see that all the guides were detected in the library. So here's an example of a bad screen, where at the end, about half of the guides were not detected anymore. And if they're not detected, we can't calculate a depletion value from that. So if you were to pick things that were depleted, I can tell you right off the bat that 50% of your library was depleted, and you're going to have a really hard time picking high confidence hits when half your library is not there. So you have about 40,000 hits. Okay, so we also look at replicate reproducibility. We look at this first at the normalized read count level. So this tells you about technical reproducibility of your screen. Not did the screen work or not. This just tells you, did my replicates perform the same way? Okay, so we see here, this is a nice example of a screen with high replicate reproducibility between 2 different replicates. An important thing to keep in mind is that this metric is heavily influenced by library representation. So things that are very, very abundant in your library, even if they're strongly depleted, may still have a lot of reads at the end of your screen. So what I've highlighted here is the common essentials that we use. These are basically proteasomal, spliceosomal, or ribosomal genes. You can't see it that well in this plot, but these points are peppered kind of throughout this whole distribution. So it's important to keep in mind this particular metric is not one that measures enrichment or depletion, but just technical variability. So to measure enrichment depletion, we actually calculate the reproducibility of log fold changes. So this is, for example, as compared to an early time point. So this is two samples looking at log fold change relative to how abundant they were in an early time point. And we see, again, this is nicely correlated. And the log fold change specifically is just the normalized score in the sample or an endpoint read versus an early time point. Or we can actually even use pDNA, like the pDNA that we use to make the virus. Okay. This is a direct measurement of enrichment or depletion. So if we color in here the guides that target the common essentials, you can now see, so here's the 0 point. All of these are kind of have some on the spectrum of depletion. And the log fold change, this is really more of an evaluation of the reproducibility of your phenotype, where things reproducibly depleted. Okay. Another important thing that we look at is depletion relative to representation. This one may not be as straightforward, but it's also very important because poor representation can impact enrichment or depletion. So you'll see this line right here going down. It's a very fixed line because you can't deplete anything. If you have 2 reads and you go to 0 reads, you can't deplete more than that. So things that are very low abundance can be very lowly depleted. So you lose your dynamic range to detect depletion. And also, think about something going from 10 reads in your starting point to 1 read at the end point. It's a big change versus 1,000 reads in your beginning point and 990 in your end point. It's actually not that big of a change, even though it's the same difference in read counts. So higher starting representations can increase your ability to accurately quantify depletion. So you'll see here again coloring the common essentials. They're depleted here, and you can see they were pretty high abundance in the starting library, so we can be confident that the depletions are actually real. Okay. Any questions? Okay. Lastly, I just wanted to come back and touch on this, lastly for negative selection screens, is the importance of using the right amount of template genomic DNA. So here we're looking at replicate reproducibility from your log fold changes, and these things are depleted. This was a screen that had an input of 280 micrograms of genomic DNA. If we were to do the same analysis just looking at 10 micrograms of genomic DNA, the exact same data, the exact same DNA, this is what the data looks like. So we're not sampling enough from the genomic DNA or from the cells that you screened, so you don't really have a good distribution, or you don't have this tight distribution around 0. It's kind of spread out from things that you never captured because you didn't sequence enough. So just to highlight the importance of using the correct amount of genomic DNA. Okay. So positive selection screens. So these behave a little bit differently. We still care about overall library representation, but we care about this specifically prior to selection or an early time point. And we also care about reproducibility, but it's a little bit different metric for reproducibility, a little more qualitative. I'm looking for the enrichment of guide RNAs. So this is an example of what guide detection may look like at the end of a stringent positive selection screen. So the early time point, everything was there. At the end, there's actually not a lot. And this gives us actually quite good signal because there's nothing hanging around. Only things that made it through your selection are still there. So this is okay in positive selection screens. It's actually great. Okay, so this is an example of replicate reproducibility in a positive selection screen. It looks pretty crummy if you were thinking about what or considering what the negative selection screens look like. So we typically see this. We're looking at just normalized read counts here. So most of the things fell out, so everything's kind of hanging out at 0 and 0. And you see there's some guides that make it through in replicate 1 only, or replicate 2 only, and then we see this area up here. So this is what we're really, really interested in. These are guides that made it through in both replicates. And if we label what those are, we'll say gene A. So this particular screen was designed around gene A. So that's very promising that was the major hit. But if we go down, there's some other things we should look at. And I mentioned it's more qualitative, because if we look at what guides were enriched in each individual replicate, you'll see here that 2 guides targeting gene B were enriched in this replicate, and then 1 was highly enriched in this replicate. And likewise for gene C, we see 2 guides highly enriched here and 1 here. Now, we didn't see the same guides enriching in both replicates, but we do see that guides targeting a common gene were enriched in one or the other. So that's why I mentioned this is a little more qualitative, because you're not going to get a fixed number for an enrichment, but you do have some confidence that was actually enriched. And then we also see things that are kind of hanging out down here a little more, where they're not quite as enriched up here, but they did have some significant enrichment in both replicates. So these are the kind of things that we'll be considering when we look at a positive selection screen. Okay, so that kind of wraps up our cursory analysis of figuring out if your screen kind of worked. And now what you're really interested in are what are my hits. So Mundra is going to come talk about guide to gene level analysis and picking hits.
Thank you. So hi, I'm Mudra, and I will be talking about how you can go from your guides to gene level. So as Scott explained, you get back your sequencing, and you have a file with raw sequencing read counts for each of the guide RNA in your library for your reference sample and your test sample. And your reference sample can be anything such as pDNA, early time point, DMSO, your untreated drug arm, and so on. So you then log normalize your data to get what is called the log norm file. So you again have log normalized values for your reference sample and your test sample. And since now you're in log space, you can just subtract the test and the reference sample values for each of the guide RNAs to get what we call log-fold change. So for these guides, you can see a negative log-fold change, which shows some sort of depletion, and for these guides in this example, you can see positive log-fold changes, which show some sort of enrichment. There are a lot of different metrics that you can calculate for each of your guide RNA in your library to rank them, and we always use log2-fold changes. You can then sort the scores that you've calculated, in this case log-fold changes, to analyze your guide level hits. And shown here from a figure taken from a publication from the Sabatini lab. On the y-axis is your log-fold change scores, and all of the guide RNAs in the library are sorted here. And on the right, you can see all of your enriched guide RNAs, and here are your depleted guide RNAs, and in green are the control guide RNAs. So you can use guide RNA level data for the initial assessment of your library, but what you really care about are your gene level hits. So again, there are so many different ways you can calculate a gene score for every gene in your library, and shown here is a table taken from, again, a publication from the Sabatini lab, where the first column shows different publications, and the second column shows all of the cell lines that were screened. So there are so many different screens that we're looking at here. The third column shows the sgRNA library that was used in each of these publications. And the fourth and the most important column here shows how gene scores were calculated for each of these screens. And you can see that in one of them, they used just the average log-fold change of all the guide RNAs. Some of them used algorithms that calculate some sort of significance, and then some of them just use the log-fold change average of the top 3 guide RNAs. So once you've calculated your gene score in using some metric, you can then again plot these scores by sorting them and look at your gene level hits, as shown here in these 2 plots, where the y-axis is some sort of gene score that was calculated, and your x-axis is just gene rank. So you can also calculate, as I said, p-values that using various statistics that can be used to evaluate your gene significance. And you can also then plot that, sort them and plot that to look at your gene level hits. Again, y-axis here is a p-value calculated using a statistic, and x-axis is gene rank. So at GPP, we developed 2 different methods to calculate gene p-values. So method 1 is uses the binomial distribution and is called stars. Method 2 uses the hypergeometric distribution, and I will be talking about both these methods now. So method 1 uses binomial distribution and is called stars. You can go to the link here in order to download the Python code to run this analysis, and it also includes the documentation that will help you run this code. So what input files you need to analyze your screening data using these methods? So the first file you need is a list of all the perturbations in your library and log-fold changes or some sort of metric to rank these guide RNAs and to calculate gene significance. So as shown in the example here, in your first column, you have all of the guide RNAs that are in your library, and each consecutive column are log-fold changes for all of the samples in your experiment. Another important file that you need to analyze your screening data is a file that maps all of the guide RNAs in your library to their gene targets, and we refer to this as the CHIP file in GPP. And if you go to this link here, you can download CHIP files for libraries that are available to the BroadE community and others. So there are two different CHIP files based on the ways guide RNAs are mapped to genes. So the first one uses a high stringency alignment where you have your 20 nucleotide sequence, And this was designed to target a particular gene. For example, here, sgRNA1 was designed to target gene A, sgRNA2 to target gene A, and so on. And these are known as your intended gene targets. So what we first do is you have your genome, and we just map this 20 nucleotide sequence to the genome, and this gives you your 20-mer chip file. The second kind of chip file involves a low stringency alignment, where you, again, have your guide RNAs in your library, and we ignore the first 3 nucleotides of each guide RNA because from previous experiments, we have found that these do not contribute as much to Cas9 specificity. So once you ignore the first 3 nucleotides, you can align the 17 mers to the genome, and now you can see that these 17 nucleotides actually align to more than one gene. So for example, sgRNA1 now targets gene A and gene B, sgRNA2 targets gene A, gene C, and gene D, and so on. So you do this for every guide RNA in your library, and what you end up with is called a 17-mer chip file. And this is the default chip file that we use for most of our analysis. To make this more clear, if we look at just 2 examples of guide RNAs from a certain library, this guide RNA here, its intended gene target was hist1h2BE, which is what you will find in your 20-mer chip file. But when you look at the 17-mer chip file, you will find 12 more targets for this particular guide RNA. And as you can see, all these genes start with hist1h2, which suggests that they belong to the same gene family and they have very homologous sequences. And similarly for this second guide RNA here. So once you have your input files, you can then move on to using these methods to calculate gene p-values. And so STARS here uses the probability mass function of a binomial distribution to calculate the gene p-values. And the formula for the PMF of a binomial distribution is shown here, where in this formula, P is just the rank of the particular perturbation you're looking at divided by the total number of perturbations targeting a gene. Q is just 1 minus P. N is the total number of perturbations targeting a gene, and K is the rank among gene-specific perturbations. So let us now look at an example to calculate a star score. So if you want to calculate the star score for a gene that has 5 perturbations targeting this gene in an entire library of around 8,600 perturbations, you have your input file with the first column having the list of guide RNAs, and your second column is your sorted log-fold change scores. And in this case, they have been sorted from the most positive to the least positive. And the boxes here in purple are all of the guide RNAs that target this gene for which we're trying to calculate the star score. So what you then go ahead and do is pick a hit threshold. Here in this example, we've picked a hit threshold of 5%, and this divides your entire input list into hit guide RNAs and non-hit guide RNAs, or basically signal and noise. And this is a user-defined hit threshold, so it's sort of an arbitrary number that you can pick. Once you've picked your hit threshold, you then start calculating probabilities. So you have your perturbation 1 that targets this gene, which is ranked 3 here. So you use that information and plug it into the formula that I just described, and you can use that to calculate the probability of one perturbation ranking 3, given all of the other perturbations. And then you do the same thing for perturbation 2, which is ranked 27, plug it into the formula and calculate the probability. Then do it for perturbation 3, which is ranked 424 here, use the formula and calculate the probability. And since these 2 guide RNAs do not fall within the hit threshold, we ignore them and do not use them to calculate a star score for this particular gene. So then you have 3 probabilities for a gene. You then sort them from the most significant to the least significant, and then take the negative log 10 of the value of the most significant one, and that gives you your star score for this particular gene. And you should also remember that every gene needs to have at least 2 perturbations that score within this hit threshold in order to get a star score. Okay. So, of course, once you do this, every single gene gets a score. But how do you know if your gene didn't score just by random chance? In other words, saying, was your screen just a random number generator, or did genes actually score as significant hits? So in order to do that, you have all your guide RNAs in your screen, and these are the ranks of these guide RNAs in your actual screen. So you run stars on that. And then what we do is just randomize the input list over 1,000 times. And each time you randomize your input list, your guide RNA gets a new rank. And then you run stars each time you randomize your input list, essentially generating what we call a null distribution. So then you compare the values you've gotten with your actual screen to your null distribution, and this helps you calculate your false discovery rate and sort of assign a significance to your gene hit. So that was stars, and you get an output file for stars that looks somewhat like this. And in the first column is the gene symbol, and as I said, not all genes in your library will be in this file. Only those that had at least 2 perturbations scoring within the hit threshold that you picked will be in this file. So your first column is your gene symbol. The second column tells you the total number of perturbations that targeted this gene. The third column gives you the actual ranks of the guide RNAs. The fourth column tells you the guide RNA from which this star score was picked. So from our example, the second guide RNA gave us the probability that we picked as a star score, so this column will have 2 for that gene, for example. This column gives you the actual star score, which was the negative log of the probability that we calculated. And the average score is just the negative log of the averages of all the probabilities that we calculated. The p-value FDR and the corrected FDR is calculated by using the null distribution. So these give you the significance of your gene hits. Okay. So the second method is uses the hypergeometric distribution, and we were just looking for alternate but very complementary approaches to analyze data, and we also this time had some emphasis on visualization, which I will show you later. So for the hypergeometric distribution, you again have your sgRNA library, and you have some sort of score to rank your sgRNA library. And say we want to calculate the p-value for gene A, which has 4 guide RNAs targeting it, shown here in red. So what you first do is you rank your guide RNAs from most enriched to least enriched. And so you have your ranks, and you get 4 different ranks for each of the guide RNA targeting gene A. And then you rank your sgRNA library in the opposite direction, that is from the least enriched to the most enriched, and you get 4 different ranks for the same guide RNAs in targeting gene A. So if you calculate the p-values using the hypergeometric distribution, you get descending p-values and ascending p-values, and shown here is just the negative log to the base 10 of p-values, so 4 values in the descending direction and 4 values in the ascending direction. And we take the average of these p-values. So now this particular gene A has 2 different p-values, and how do we pick one value for this gene A? What we do is just pick the most significant one, and this gets assigned as the p-value for this particular gene. And you do this for every single gene in your library, so each gene ends up having a p-value. Okay. So method 2 was also implemented in Python, and if you go to this GitHub link here, you can download the code and run it. And the output file for this method looks like this, where the first column is your gene symbol, and please note here that every single gene gets a p-value. The second column here is the average log-fold change, which is just the average of the log-fold change of all the guide RNAs targeting this particular gene. In the third column, you have your average p-values, which we just calculated. In the fourth column, you have the total number of perturbations that target this gene. In the fifth, you have the sequences of the perturbations themselves. You have the individual log-fold changes for each of the gene symbols. You have their ascending ranks, ascending p-values, descending ranks, and descending p-values. Okay. So when you calculate, when you run each of these methods on the same screen, one of the screens I picked was published by the Waldorf lab last year, and I ran them using both methods, and if you compare the p-values, how do they compare? So shown here on the x-axis is method 1, p-values, and y-axis is the p-values for method 2, and you can see that they line up really well with a Pearson coefficient of 0.84, and all of the top genes remain the same, whether you use method 1 or method 2. So this sort of suggests that most algorithms will end up giving you very similar hit lists, so you do not have to analyze your screen using every single algorithm that exists. But having said that, you should definitely analyze your screen with more than one algorithm because of this fuzzy region here, which can be slightly different depending on what method you use. So try to tease out as much information as you can from your screen, so use multiple methods. Okay, comparing these two methods further and looking at some of the differences. In method 1, you have to run the code 2 times to identify positively enriched genes versus negatively enriched genes, so one sorting in the positive direction and the other time sorting in the negative direction. But with the second method, you can get hits in both directions with just a single run of this algorithm. In method 1, FDRs and p-values are assigned, as I said, to only genes that have at least 2 perturbations that rank within the hit threshold that you picked. And whereas in method 2, every single gene gets a p-value. In method 1, you have to generate a null distribution, which can be very time-consuming, so that takes a little longer to run, whereas for method 2, you do not have to generate any null distribution. So from our guide level data, we have now calculated the magnitude of a gene, which was just the average log-fold change of all or some of the guide RNAs that target this gene, and this was what you actually measured in your screen. And then we have also calculated gene significance using 2 different statistics, using the binomial distribution and the hypergeometric distribution. And so now let's look at how this can be best visualized. You can visualize this data just by looking at standard volcano plots, where on the x-axis you have your average log-fold change, and on your y-axis you have your average p-values that you calculated. And shown here in gray are just the non-targeting controls, and you can see that they line up around 0 in terms of fold change and also in terms of p-values, so showing that they don't have much of an effect in the screen, which is what you would like to see. And as you go towards the right on the fold change side and also upwards on the p-value side, you see these genes that are enriched in the positive direction in color here, and their gene symbols are mentioned here. And similarly, in the opposite direction, you see most depleted genes with higher p-values in color here, and their gene symbols are shown here. So this is a very good way of visualizing your gene level data. There are so many other tools out there that you can still use to look at gene level hits. And one such tool is called MAGIC. and we definitely recommend using this tool. This stands for Model-Based Analysis of Genome-Wide CRISPR-Cas9 Knockout. It uses the robust rank aggregation, which is a method very similar to those that I just described. And this can be run once to identify both positively and negatively enriched genes. And what you need as input is just read counts of your sample, but I think this algorithm needs you to have multiple replicates or time points in order to analyze your screen. The second one mentioned here is BAGEL, which stands for Bayesian Analysis of Gene Essentiality. This uses a Bayes factor and trains sets of the essential and non-essential gene sets to determine gene essentiality of every single gene in your library. And this also uses read counts for each of your sample as input. And the third one mentioned here is RIGOR, which stands for RNAi Gene Enrichment Ranking. So we definitely don't recommend using this for analyzing CRISPR screens. This uses the KS test, and it has a low sensitivity in identification of gene hits. But if you still decide to use it, then you can use any metric that can be used to rank guide RNAs as input. So now that you're thinking about analyzing your data, what are some things that you really have to pay attention to when you're doing some data manipulation or data handling? You should know that Excel converts some gene names such as March1, Mark1, or Sept1 into dates. So please make sure that these aren't changed to dates before you save any file. And then if you are doing a lot of sorting of your data, please make sure you select all of the columns in your Excel file before you sort them. Otherwise, your data can get shuffled, and this can lead to a lot of problems further on. And also, when you're using two separate files, if you are copying, pasting from one file to another, please make sure they're sorted in similar ways before you copy and paste from one file to another. And another thing you should just know or remember is that gene annotations and symbols can change all the time. So if you redo your analysis, say after an year, and you see new gene symbols popping up, just don't panic. It's just the way human genome annotation is. All right, so now you have your list of hit genes. So how do you interpret your gene hit lists? So one tool that was developed at the BroadE for pathway analysis is called Gene Set Enrichment Analysis, for which the link is here. You can run this essentially in two ways. You can either download the software onto your system and use that, or for a quick overview, you can click Explore MSigDB, where MSigDB is a database that has all of the gene sets that you can compare your gene hit list against. So once you click this, it takes you to a page where it shows all of the gene sets that are available, and then you just click on Investigate Gene Set, which takes you to this page where you can paste your list of gene IDs or gene symbols to analyze, and then you select all of the gene sets that you want to analyze it against, and then click on Compute Overlaps. It gives you a result where it gives you all of the pathways that are enriched in your gene list, and then also assigns a p-value and FDR to them. If you end up downloading the data and analyzing your hit list that way, it also helps you generate these beautiful enrichment plots for each of the pathways that your gene hit lists are enriched in, or the ones that you select to look at, and it also gives you an enrichment score for each of the pathways that you can use. And so this is one tool to use to analyze your gene hit list, but of course there are so many other tools out there, and 4 tools are listed here: String, Panther, David, and Reactome. And each of these, you can use your gene symbols, your gene IDs, etc., to query using these tools. And each of these tools generate these functional associations between each of the proteins that you're looking at and assign some sort of score to them, so you can use that for further analysis. And one thing that I've noted here is how frequently each of these are updated. And these functional annotations can change, and so please make sure that the tool that you're using has updated them very recently before you start and analyze your gene hit list with that. All right. And John will be continuing. Thank you.
Any questions on Mudra's stuff? Okay, great. So final bit before we end, and I'll have lunch. I just wanted to mention that, at the end of a screen, one thing an analysis can't do for you is tell you what to do next. You can get lists of genes, and you can say, aha, this gene scored. But rarely does the identity of that gene in a hit list tell you what the hypothesis is. Usually it's a gene you've never heard of. Likewise, if at the end of a screen, the top 10 genes, you look at it and say, oh, I knew those were going to score anyway. Well, then what was the point of doing the screen? You didn't learn anything. So, the entire morning has been about, how do you set up your assay and how do you execute it right and how do you analyze it right. But the most important thing before doing any of that is to figure out and to really state for yourself, why am I doing this screen in the first place? Or maybe another way of saying that is, what are the experiments I'm going to do in small scale that I can't do genome-wide? Once I find that a gene is implicated in this phenotype, why do I care about it? What am I going to do next? What is the abstract of my paper going to say? Make sure you know that before doing a screen, because you can get stuck in having a hit list, but then not really knowing what to do. And no amount of validation of saying, well, okay, yeah, put in more guides, and yep, I've confirmed my phenotype, and yep, I've done a rescue experiment. That's not really moving your project forward. You need to know, why did I do this screen? What are the experiments that's going to come next? How am I going to make a story out of my screen? So I want to give just one final example of a screen done here at the BroadE a couple years ago from a Viv Regev group that I thought was just a great example of going from start to finish of a screen in a very challenging model system. So this was done by Oren Parnas, where they started with the Cas9 mouse. They harvested primary cells out of the mouse and then delivered a genome-wide library, did an in vitro assay, and then put the cells on the flow cytometer, and we're looking for intracellular TNF levels. So they fixed the cells, stained them for TNF, and then they sorted, well, what are the cells that have high levels of TNF versus what are the cells that have low levels of TNF? Okay, so this was their primary screen. Very challenging. They had to sack a lot of mice to do this. And this is what their replicates look like coming off of the flow cytometer. Here's the log fold change for replicate 2 versus replicate 3, and it was a complete hairball. Now, I should point out that this is actually not all that uncommon when doing flow cytometry-based screens. Flow screens are very challenging. There's something you probably don't want your first screen to be a flow screen. It's something you want to work up to. But this is what they got. They got a hairball, but not all was lost. So here I generated volcano plots, just as Mudra showed previously, for the 3 different replicates, and you can see, okay, some genes do score. I think it's not a coincidence that replicate 1, shown in green, is the flattest. Then replicate 2 starts to look a little better. And then replicate 3 looks the best. They got better at doing the assay each time they did it. And thus they got better data. They got better guide-to-guide consistency. So even though the primary data was a hairball, there is some enrichment. Maybe another way of saying that is there is some enrichment for particular guide RNAs in this quadrant and particular guide RNAs targeting the same gene in this quadrant. Okay, but then the next step, and I think this is a really generalizable step for a lot of the next thing in a pooled screen, is they made a custom pool, where they now, for all the genes that hit, whether they scored strongly or whether they scored very weakly, like maybe they were hit, maybe they weren't, they made a custom pool with many more guide RNAs per gene, and then repeated the screen. And now when they did this, because they could get far higher representation, because their library was much smaller, the secondary screen and assay was very, very clean, and they get really, really significant p-values from their screen. And you can see that in this heat map on the right here, where the first 3 columns are replicates 1, 2, and 3, and the p-value of the different genes. And you can see there are some genes that scored in one replicate and didn't repeat at all in the secondary screen, and there are some that scored somewhat heterogeneously in the primary screens, but then repeated very, very well in the secondary screen. So this is a very good generalizable strategy for following up on a primary screen where primary screening data can be noisy. But then if you make a much smaller library, you can do a lot better. But what's interesting is here I'm plotting the p-value from the original screen and then the validation rate in the secondary screen on the y-axis. And what you can see is a very nice correlation between the reproducibility or the validation rate based on the p-value in the primary screen, which suggests another generalizable strategy is that, so you're going to perform your primary screen at large scale. For the top genes that score, immediately begin your follow-up on those. Perform the focus assays that will start to show you, well, what were the blind spots in my assay? What was this assay really scoring for? Was it reproducing, or was it representing what the biology I wanted it to have, or is there some artifacts coming through in here? And then also, we're all human here. Just by actually doing some of these focused assays, that often tends to stimulate thoughts about what additional follow-up assays would be. It's a way of forcing yourself to engage with telling the story earlier on in the project and saying, well, okay, what do I care about these genes? But then in parallel, launch the creation of a custom pool. So again, target far fewer genes, but more guide RNAs per gene, so you have more statistical significance. But since it's going to be a much smaller library, you can then take that secondary pool and screen it in more cell lines, screen it in more conditions, maybe even think about doing an in vivo screen if you had done an in vitro screen previously. So having one of these focused libraries at the end can be really, really valuable for really helping to move your project forward and have a very clear hit list when you're done with your screening project. Okay, so just to wrap up, how do you get started with GPP? So generally the way that most projects start is you'll meet with a screening scientist. We actually have reserved every Monday an hour and a half where Dave Root, myself, Federica, and Scott all have that time blocked off. So it's very easy to just email Olivia or one of us and schedule a time then, and we'll meet with you and talk about your screen for an hour and a half. It's great if you bring preliminary data, and I don't even mean preliminary screening data or preliminary assay dev data, just here's our system, here's the question I want to ask, here's the cell lines we're thinking of using, here's relevant examples from the literature, whatever it is. And that tends to be a very fun discussion for an hour and a half of how to start moving your screen forward. And 3 out of the 4 people here are actually quite nice. Not me. You then generally, as I mentioned, you can obtain a lot of reagents from us. We have a website. I'll show you the link on the next slide, but we have a lot of reagents that will help get you going so that if there are things that, as an example, if you've never prepared lentivirus before in your life, you don't need to spend 3 months figuring out how to make good lentivirus. We have it in the freezer and you can get going. Now, obviously, if during that time you want to become expert in preparing lentivirus, great, work on that, but make sure you're moving your screening project along at the same time. Then during the screen, as several of the talks mentioned, there's a lot that can go on during a screen. A lot of preparation, where it's really good to interact with your screening scientist and say, hey, here's what my data look like. What do you think? Does that look good? Does that look bad? And since we've seen a lot of screens, we can generally recommend, we have a sense of, yes, this is in the range of normal, or, oh, geez, you might be, encountering a problem like this. So to really have a good back and forth with your screening scientist while the screen is ongoing. And then as Scott mentioned at the end, this is a step that I think is particularly useful for us to perform. You can just send us a plate of genomic DNA, and then a couple weeks later, you'll get back data and some preliminary analysis for you to start moving forward with. So we have a lot of reagents at the GPP. We have a lot of vectors, whether it's for shRNAs or ORFs or sgRNAs. Many of these things are in inventory. So if there's a particular ORF that you're interested in or a guide RNAs targeting your favorite gene, a lot of times it's already in our freezer and you just need to get it from us. So you can save yourself several days or weeks of cloning just by pulling it from our inventory. But if there's something that we don't have, we can make it for you. So, for some people, the idea of cloning 90 guide RNAs in a plate is totally trivial. For other people, that's terrifying. If you're in the terrifying camp, we're happy to do that for you. Likewise, we can make things as individual constructs. We can make things as pools. And we can provide glycerol stocks. We can provide prepared DNA, or we can provide lentivirus. So we're really happy to work with, with you for whatever is most necessary for your screening project. So if your PI, if you have a BroadE email address, then you already have access to our website, but just by logging on to the BroadE intranet Wi-Fi. But if your PI is a BroadE associate, but you don't have a BroadE email, you can email Olivia to get access to our website. We have a public-facing website, but the intranet site has everything that's on the public website and more. We don't try to be exclusionary, but we can't support the entire world's screening efforts. We focus on the BroadE first. So there's more information on the BroadE intranet site than there is on the external website. And again, if you don't have a BroadE email address but want website access, just email Olivia to get it. Yes, contact Olivia. Okay, with that, I want to just close by thanking everyone in GPP. We're a group of about 35 folks or so that are really focused on doing these screens and trying to just scale this process as much as possible across the entire BroadE community so that your screening project can be a success and you can really focus on the biology of why you did the screen in the first place. So with that, we're ending a little bit early, which is great. As I mentioned, we'll hang around up here, so if you have any further follow-up questions, feel free to come up and ask. But otherwise, thanks so much for attending.